and I'd like to introduce Thomas Landrin, speaker of JOGL from France. He will carry a 3-hour block discussing all the innovations that JOGL has been taking part in throughout the world over these past few years, and especially throughout with respect to COVID-19. These involve the talks of creation of an open biosafety and biosecurity framework for independent scientists, a collaborative effort by iGEM and JOGL, an update on SARS-CoV-2 diagnostics by JOGL, followed by a break. This will then be followed by a talk of novel biosafety and bioethics questions in the JOGL Open COVID-19 program by Alex Pearlman of UPenn, Rebekah Choi of UPenn, Lisa Rasmussen of UNCC. This will be followed by an open discussion on supporting OpenBio innovation to bring them to market. Introducing JOGL Fitz by JOGL. And I'll let Thomas take it away. Thank you, Thomas.
Thank you so much, Gabriel, for the great introduction. Hi everyone. I'm extremely pleased to be with you in this Ribosome Zoom room, and we're going to talk about several subjects during those next 3 hours. But the first subject is going to be quite interesting for the global community, as it's been one of the key obstacle for this community to mature and to grow, as it's been targeted on specifically biosafety and biosecurity many times. And so when we came together with iGEM Foundation, has an extensive experience also on dealing with those issues with the iGEM community. It was a very natural fit to create a service to the open community to help with managing those biosafety and biosecurity issue by just helping every project to get reviewed and to have a better sense of what are the best practices to follow. So I'll go ahead and share my slides. Is this new? Yeah. Is that how I go full screen? I guess so. Yeah, perfect. So this is about the creation of an open biosafety and biosecurity framework for independent scientists. And as I was explaining, this is a collaborative effort between the iGEM Foundation and JOGL. And I would like to invite Sarah Ware, who has been really a key member of this team behind this project, to actually introduce the project. Sarah, are you with us?
Yes, can you hear me?
Perfectly.
Great. Hi, yes, so we're very excited to do this presentation for you guys. So as Tomas mentioned, we have JOGL, we have a collaboration now with iGEM, and this is to create a framework for a biosafety and biosecurity review board. And so why do we have this initiative? Well, as we'll touch on later too, who in this community wants to be doing unsafe work? I hope none of us, no one's going to raise their hand and say, oh no, that's me. So we need this kind of framework so that we can have a way to assess projects and, are these safe, or could they be safer? So we'll talk about the main objectives, and along those lines, one of the main points is that both iGEM and JOGL have a history of supporting biosafety and biosecurity for independent projects. So Tessa and Tomas will talk some more about that. We need this framework to work for the entire community. And by the entire community, I mean all of us listening to this, all of us that are registered for the Global Community Bio Summit, all of us who are doing independent research. we need this kind of framework. And so although iGEM has this kind of framework and JOGL has this kind of framework, we need it to work for the entire community. So this is the goal. There is already grant money earmarked to develop such a framework. So this is wonderful. So Tessa will talk some more about that. And then we will let you guys know about our timeline and scope. So this is not far off into the future. This is being worked on right now. Tomas, if you could go to the next, thank you. Okay, so why do we need this? So guidance is largely assuming that there's an institutional lab environment instead of somebody's shed or house or community lab or workshops, but we need this kind of guidance for our community. So the biosafety and biosecurity could be strengthened for all of our projects if we had the relevant support for this and we had reference materials. And so one of the things that we aim to do is to create a reference bank of really great projects that are really taking into account biosafety and biosecurity that can be referenced by everybody as examples. So we hope to create, to foster a lot of these really quickly for the community. We need to have this set of references again so that people can access them, can see what a good project is incorporating, can adapt it for their own projects. And then we want to incorporate, which is very important, we want to incorporate community peer review. This is already done within JOGL. So within JOGL, just one giant lab, there are many grants available periodically. And there is an evaluation process. So there's a very specific submission process that's pretty formalized at this point. There's an evaluation process that's pretty formalized at this point for projects in general, should they be funded or not in the community. We do this through community peer review. And so we want to also include community peer review within this biosafety and biosecurity framework. Tomas? We want to increase the biorisk awareness and management in the OpenBio community. So even by the creation of such a framework, this is going to increase awareness that, hey, we need to be taking these things into consideration, and even though we are doing independent research. So we also want this to be a very public process. So we want this to be very transparent in how we are evaluating these projects. And again, as mentioned, we want to set up this reference bank. And we want to provide a digital infrastructure. JOGL already has this model, so that we're going to be needing to create these submission forms. These submission forms for biosafety and biosecurity review will be different from the submission forms for general projects on JOGL. So these are going to be asking questions like, what are your safety procedures? What kind of organisms are you using? What kind of equipment are you using? What kind of training are you providing to people in your lab or in whatever space that you have? Okay, and allowing projects to be triaged based on peer review. So what we mean by that is that projects can be submitted and peer review, your colleagues are going to take a first crack at it, let's say. And those projects that pass the peer review process will go on to be evaluated by some experts, not necessarily peer review. Okay, as I mentioned earlier, so the why do we need to create this framework? Who of you wants to do dangerous work? None of you, right? We want to make this free. We want to make microgrants available. We want to provide some incentives for consideration of biosafety and biosecurity in our community projects. This could also be a protection somewhat for bad PR for the community. So if there is a project from the independent science community that is not falling within guidelines for biosafety and biosecurity, at least we as a community could say, you know what, we had this framework and these people opted out. Okay, so then awareness improvement by learning through reviewing projects of peers. Even just, you know, I'm a professor, and so sometimes I have students review their own papers before I get them. And in part, it's this idea is that if we are reviewing other people's projects, it's also going to make us more aware of maybe some things within our own projects that could be improved. And then creating this library of validated projects so that the community can use those. Okay, so at this point, I would like to turn it over to Tessa and Tomas to talk about how iGEM and JOGL both already have a history of supporting biosafety and biosecurity for independent projects. So very exciting.
Yeah, so I'll jump in and we can hop over to the next slide. Ooh, I think we maybe hopped 2 slides ahead. Or the slides are out of order. That's fine. I'll just present from this one. So we are jumping ahead, I think. All right, so I wanted to talk a little bit about, you know, some of the ways that we think about risk assessment in the context of iGEM. And, you know, I think talking about some of the stuff that Sarah was saying, where, you know, a lot of these frameworks assume that that people are operating in a sort of like formal lab environment that maybe has like some kind of biosafety officer, or maybe there are like, you know, strict regulations defined by the local government that determine what you're allowed to do in that kind of space. But we, within the iGEM competition, have a lot of people who are not working in that kind of environment. Both we have people working in community labs, and we also have folks working out of, you know, commercial labs, which are often run pretty differently from institutional labs. We have high school students, including sometimes high school students in our experience working with like fairly nasty toxins. So I think we, you know, in the iGEM safety and security program, which is where I work, you know, our sort of step 0 is always like, all right, make sure nobody gets hurt from their iGEM project, right? That's kind of the bottom line. But then I think beyond that basic risk assessment, which is about make sure that nobody's immediately getting hurt, there's some other aspects of risk assessment that I wanted to touch on as part of this discussion, which is that, you know, with our safety and security program, you know, I think we're also trying to set up and reflect some community values. So, you know, one of the aspects of that is that within the iGEM competition, we ask everyone, every team to do a risk assessment of their project. And that's really trying to communicate this idea that risk assessment and thinking about biosecurity and biosafety isn't somebody else's job. You know, that as a synthetic biologist, that should be one of the things that you're interested in and taking some time to think about yourself. And I think we sort of feel like maybe we have an opportunity to dialogue with folks and set up some of those community values, because for a lot of people, iGEM is their first or very early experience with synthetic biology. So with programs, not only our safety program, but also our human practices program and some of our diversity and inclusion stuff, you know, we're hoping as a community to really like nurture values of respect and responsibility and thoughtfulness and reflection. Next slide, please. And, you know, I think thinking about those values and thinking about like as a community figuring out a good future, you know, people talk about these decades of synthetic biology. And so, you know, now we're in the 3rd decade, unclear what the theme of it will be, but, you know, at iGEM, we're kind of hoping that the theme becomes, hey, like people are empowered everywhere in the world to solve their problems using synthetic biology. And then maybe we get towards the cool like solar punk features that are in Black Panther and some of the really cool solar punk art. But, you know, among many things that need to happen before we can get to that future is innovation on biosecurity and biosafety. You know, I think as a community, it's not like we're owed trust by people that what we're doing is safe and reasonable. I think that we need to work on demonstrating our commitment to taking into account stakeholder feedback, demonstrating our commitment to really doing a reasonable risk assessment of our projects, and doing so in innovative ways. So jumping over to the next slide. You know, iGEM's in the midst of like trying to support some technical innovation in that. So that's, you know, providing grants to some teams, creating a safety and security award. This year in 2021, we had the winners of that award were working on things like increasing the ability to do cell-free work in plants so that you could have kind of minimized risk of horizontal gene transfer while still being able to do plant synthetic biology, or innovative biocontainment systems that use interesting hydrogels, or sort of full of project safe by design analyses, and doing those in a way that other teams might be able to copy them in the future. So there's that kind of that world of technical biosecurity, but then I also think my personal belief is that we also need innovation in risk assessment, which is why I'm so excited about this JOGL iGEM collaboration. So if we jump to the next slide. Interesting. I thought there was another slide about iGEM stuff, but whatever. Yes, great. Community-based learning. So this is really, you know, if I have found that there is not a huge consensus about like, yes, we know how to assess the risks in biology projects. Even if you ask people what kinds of risks are they worried about from biology, you will get different answers. And so that's something that we've been looking into this year. We did this risk assessment study that we're still analyzing the data from. So no conclusions for you yet, but really just asked a wide group of people in our iGEM community to look at some past iGEM projects that were anonymized and just ask them a bunch of questions about how big are the benefits of this project in your mind? How risky do you think it is? You know, what sort of measures would you suggest to mitigate the risks in this project if you see any, or if you think any of them are necessary? And what I'm really curious about there is there a consensus? You know, do people perceive the same risks in the same projects? And my guess is going to be that the answer is no, people don't perceive the same risks, but I don't know yet, which is why we did the study. But I think that kind of, you know, crowdsourced community intelligence about how to do this kind of risk assessment is a really promising way to learn how to do it together. And so, you know, this upcoming JOGL collaboration, I'm so excited about it because I think we can learn from each other by building this repository of really like well-validated risk assessments that we'll be able to use as a model going forward. So that's my little spiel about how this sort of fits into the larger way that I'm thinking about, you know, as a synthetic biology and an open biology community, like how do we get better at risk assessment? I think this collaboration has the potential to be like a really cool part of that. And I will pass it off to Thomas now.
Thank you so much, Tessa. To go on Tessa's introduction to what the iGEM Foundation has been doing regarding biosafety, we don't have the same level of history as iGEM Foundation, obviously. What we did, however, was to actually create in a community setting biosafety and biosecurity guidelines for the Open COVID-19 initiative, and that is something that appeared very early in the process of the creation of this community. In fact, in the very first meeting that gave birth actually to this initiative was raised biosafety and biosecurity guidelines, and so we decided to create a board of people who had experience working with biosafety and biosecurity, and so they created this document, which is an open document that we can go and read. It's under a CC0 public domain waiver. And also we made sure that any projects that was going through the Open COVID-19 process to get supported, get reviewed and get access to grants, had to, agree by those guidelines. Like they had to work by those guidelines. And if they didn't, if they didn't comply, were not compatible with those guidelines, they couldn't actually access the support by JOGL. And we had one example of such a project that didn't make it, even though the idea behind the project was very seducing, which was the open vaccine project called RADVAC, and they were just too open about the idea of self trial. And so that was going really against our own guidelines. So we actually entered discussions with them about how we could assist them in changing their processes, their protocols and their methodologies. But it just didn't happen at the end, even though there was goodwill from both sides. But the thing is, the RADVAC is definitely not an isolated project, and usually it's not because the project leaders want to do things in a rogue way. It's just that they don't necessarily have the right support and expertise actually to understand what they're doing actually could be wrong, for example. So just to because at the end of this presentation, we're going to enter an open discussion. This is really the main part of this section. It's that we really want actually to hear from you what you think about this initiative, and so we are going to we want to hear, for example, from you, like, what do you think are good requirements for documentation, or what kind of criteria do you think experts and peers should use? We obviously have our own ideas with especially with the experience that we got from the committee and from the iGEM Foundation, but we really want again to hear from you. For the one that didn't assist to attended to my talk earlier today, this is just a summary of what is the JOGL community review approach. It's a process that was really created and tested and optimized during the pandemic, as we went through 5 cycles of reviews. And basically, you have a project that needs to document its ID and process and methodology and according to a template that is provided to them, and so that all projects follow the same template. It makes it easier actually for reviewers to go through them. Then reviewers are associated with the project, and all reviewers are anonymous, which makes the process more robust in terms of relevantness for the answers. This is based on peer review research, and then this evaluation gives birth to on one side to a score for feasibility, a score for impact. That was for the Open COVID-19. You could imagine actually different kind of score for this initiative, obviously, and it gives birth also to feedbacks, and those feedbacks are then used for the project to either improve, for example, if the score was not good enough to be validated, and that you could try again, at the next cycle of evaluation, and or the score is good enough, it's above the quality threshold, and then the project becomes validated. And this process is also very scalable because you don't only have on one side reviewers, waiting for project to be reviewed, and as an applicant, you just come and be a client of the system where you apply and then you just wait for the systems to work. This is really a distributed, way of functioning. So applicants actually are also reviewers. And so that makes it actually very scalable, because as the number of projects increases, the number of reviewers does as well. So And also because we want the process to be robust, for a project to be fully reviewed, we need at least 4 or 5 reviewers. And for a reviewer to be taken into account, they need to review at least 4 or 5 projects themselves. So this whole thing actually is what makes this committee review works, and we're going actually to publish a methodological paper about that shows how robust it is. So this is very good news, and that it actually is managing bias in a very good way, and as well as, for example, more traditional institutional way of doing reviews, like in the European Commission, for example. This is maybe a big chunk of text to swallow, but this is so far the type of modus operandi that we imagine. First, so this is going to be a digital program, meaning that everything is going to happen on the local platform. So you need to create a project page, and to document part of the project, at least according to the template that will be providing your project there. That will mean that this is becoming then openly available to any member of the community in general. Then the projects are going to be validated or not by the community peer review, or to become references for others to follow. This is also going to use the same methodology I just spoke about. Evaluations are going to be done both by the practitioners of this community and also by volunteering biosafety and biosecurity experts, as Sarah explained earlier. And so this is going to be mostly done on a double stage way, so that the members of this community are going to review all projects, and so only the best ones are going to get then evaluated by the biosafety and biosecurity experts, so that we don't overload them with projects that, for example, are not very well framed, and so it would be a loss of time for them. Everything will be published publicly, meaning that this is the way that we've done things in the past at JOGL, and it was very useful for the whole community to not only hear about the good evaluation, but also about the less good evaluation, because everybody's learning, through that. Unless, and this is also important, that we actually agree that some of the projects may need to be redacted because of dual use concerns, for example, or offensive languages. Sarah also mentioned a very important point, is that we really want that projects document their process as well as possible. And so for that reason, we're going to provide micro grants in the order of a few hundreds of euros for each project that obtain a high enough score to become references. And so this is a way to award the effort that has been put into the documentation. And obviously we'll have to work on the template, and we want the template that will be developed for both project submission and evaluation of projects. Well, that will be fully transparent, obviously, but we want this also to be validated by the community. So you'll be hearing about, from us about that in the near future. The timeline is very short. We're working actively currently on the preparation of that framework, and that is going to go until January, and we'll be launching on the earliest, the end of January, this program, probably in the beginning of February, because of the Christmas holidays. And then we have 3 rounds of evaluation, meaning that even if you actually didn't take part in the first one, you'll have actually 2 more chances. And if you actually didn't get to be validated in the first round, for example, you always be able to use the feedbacks that you got from the reviewers to improve your project and get into the second round in an improved manner. And then on our side, we'll be using all the data that we'll be accumulating from this experiment, because this is really kind of like an experiment that we want actually to generalize in the future. And so we need to do some more impact analysis so that we can come back to our funders in order to get more support. All right, this is the best part of the discussion. Thank you so much actually for listening, and I hope that you have great questions and feedback, because and the mic is open. Thank you again also to Sarah and Tessa for presenting and being part of this amazing project. All right. How should we go at this? Anyone who would like to ask a question, please raise your hand, and you can unmute yourself, or you can ask the questions directly in the chat. No questions?
Well, I hope that we would love a great discussion at this point. In general, even like, is this something the community wants and values, which we obviously think we should as a community, but general feedback, please, that would be great.
Derek?
General feedback through the process has been, wow, been great watching these projects get reviewed in public and the outputs that come out of them. And one of the things that I haven't followed as closely is how you're going about funding them. What's your funding loop look like? And does that pose project constraints? I mean, here in the pandemic, you've had an amazing focusing agent. Everybody has the same goal, as Rachel talked about in the chat. But going forward, what does funding, what does the tying of the funding agencies and the projects look like when there isn't such a clear goal?
So this is actually, there are focused funding for this program that has been mostly handled by the iGEM Foundation so far, as this is a proof of concept, using themselves actually funding they got from a couple of foundations for their work on biosafety and biosecurity. And so the goal is really actually to get enough recurring also funding. You know, if we show that this is effective, obviously, and this is a first step, and that it has a true impact on the community and the work that it does, and the way it is also received publicly outside the community. So the funding is coming from foundation, if that's what your question was about.
Well, it is, but it's more how to get the funding sources. The model that you guys are approaching here in terms of review and acceptance of projects and funding them is a much more open, accessible model to the type of science that those of us in this community do, certainly, than many of the academic funding models that academic and other industry funding models that take so much of the research dollars in this. So over time, we have to convince those traditional funders to move some portion of those traditional funds to this type of open model.
Right, yeah, you were talking about in a more general way. Okay.
Yes.
Oh definitely, yes. This is part of the JOGL agenda also, to convince those big actors to, instead of going through funding traditional institutions, you know, like creating a university chair, actually they could actually create a community program directly, you know, undergo, so that the funding is redirected to a much diverse set of practitioners, like we did with COVID-19, and so it's something that you know, I mean, this is a bit apart from the open biosafety and biosecurity framework, but we actually welcome propositions of you know, if you'd like to create such a program, you know, on a thematic that you like, and that you need to have a platform such as JOGL to back you, in terms of methodology and efficiency in the history, please do contact us, and so we can apply together, for example. Would you see yourself, this is a general question for everyone here, using this service for your own current project?
Right, this is the question. How can we make it work for everyone? How can this be something that when you first think of a project idea and draft it, what would make you want to go through such a review process? How would that look for you? Oh, there are lots of yeses. Great.
2 yeses already.
Have you faced issues regarding biosafety and biosecurity in your project in the past or currently? Or that you'd like to share? Either like maybe accidents that happened around you, or even just like doubts that you had about your own process, or maybe doubts about an initiative that you've seen in the community and you didn't know how to handle that.
Well, without, bringing up specific examples, I mean, probably a lot of us in the community can think of rogue projects. Not too many, thankfully, actually, but some. And I guess the question is, would those projects have submitted, or would they submit anyway to such a review process? But again, yeah, injecting DNA. But again, at least as a community, we would be able to say, like, these projects didn't opt to go through this review process, and our community does not condone it, so that hopefully it doesn't give a bad rap to all of us.
Home lab grown meat projects and botulism. Botulism. You've seen a project about botulism?
Or maybe this is just something that people got to think about. Maybe people haven't been thinking about this. And it's going to take a minute to be like, hey, why was this presented? Why was this important for me? Which is, part of what we want to do is raise awareness.
Derek, you have a question?
Yeah, let me ask a question around that hypothetical case of the project that as a community we really don't necessarily condone, that didn't submit through the review process that you mentioned. Is there any reason that the genesis of this discussion over risk and biosecurity needs to be at the request of the project owner? That a lot of this process, I think that there is a good structured way of having a community discussion around the risks of a project that perhaps hasn't been submitted for funding if there are community concerns about it. Is the structure that you're proposing just a consensual submitter of the project process, or are there learnings for other elements of the discussion?
This is a general process. Is not actually to get access to money. We provide a very small micro grant as a way to cover, let's say, the cost of doing good documentation. But no, this is a free service for any project that like to actually get reviewed by members of the community and by experts in biosafety and biosecurity, so that their evaluation is transparent and shows that they don't have any big flaws. So it's even if a project is not looking for money, it's a way actually to gain legitimacy and in front of the community and the public.
Thanks. I see the advantages for a project and applying. I think for most of us here on this call, we're doing projects that we'd love to have more visibility of the biosecurity and review of the project. My concern underlying it was just riffing off of Sarah's comment about somebody who was doing a project that didn't fit with our community ethos as to what our response is. And there has to be somewhere to have a discussion around that as well. And I think the process that we're talking about here around starting discussions around responsible projects has to also have reflections out on those that are not necessarily doing things that as a community we condone. And I'm not sure where that discussion happens.
Yeah, that's a really good point, Derek, actually, because where are these discussions happening? I mean, they're not for the most part, or we know of projects, but we're saying or doing nothing about it. And then again, I agree with you. I don't know what kind of space that would need to occur in or what that would look like, but it's a good point.
Eric asked, at what point do you cancel projects? So we won't be the one canceling projects. So this is the best part of this approach. This is a collective intelligence approach where the project is submitted to the community, and then it's you will have like several members of the community that will go through the description of the project. And so if first the community believes that the project looks like bad or dangerous as a whole, that's the voice of the community, and so that project won't be going to the next stage and won't be exposed to the evaluation by the biosafety and biosecurity experts. And so, however, if it's not voted out by the community because they don't feel actually comfortable with it, well, then it can go through to the second step. So and the biosafety and biosecurity experts can then point out issues with the project, and so that project won't be canceled. What will happen is that the project may actually not get validated, but they will be brought back with a lot of feedbacks to potentially improve their process. Or if it's actually something that is really at the core of the project, that actually then the project won't just be validated because it's probably considered too dangerous.
Right, for sure. I mean, this review board would not exist to say to anyone, you can't do your project. There's no governance capability for that within this community, and that's not what we're trying to also create. It's just trying to create this option for people who want to be more responsible, as Shritha put it in the comments. And so that's the reason you can't tell anyone in this community anything that you have to do this or you have to do that. This is never going to work. It's just presenting an option.
Sarah, in the case where you might have patients who hear of various projects that are available out there, and they might not have full knowledge of them or might not have oversight of medical professionals like directly overseeing it, how would they get information or be able to leverage this project to know whether something that they're considering doing is reasonable or not?
Yeah, so to bring in people who maybe want to do self-experimentation.
Or people who are being offered kind of therapies, potential therapies or offshore therapies, things like that. Do you think there's a potential there to help patients make correct, like scientifically and medically validated decisions?
That's interesting. I mean, I like the idea to open it up to kind of such a thing like that. Yeah, what do you think, Tessa, Tomas?
Actually, it somehow cut a little bit for me for the last minute. Could you repeat the proposition?
Yeah, no, it's just, you know, if you have patients out there who find out about, or people who have something that they want a therapy for or something to try, and they see or hear about these things through various different media, etc. But they're not sure whether these things are reasonable or not. If those projects themselves aren't, you know, getting validated by this project, is there something that patients themselves could actually query and find out whether the community considers this to be a realistic or reasonable tack?
That's a very interesting point.
Just a thought.
Yeah, the pipeline would probably be a little bit different from the one that we have currently imagined so far. But this is definitely interesting. Yes, we could actually see how we could adapt, you know, what we do to this for doing this. Yeah, definitely, it's a very good idea.
Yeah, nothing else on that. Oh, Tomas.
So I'm curious what you think about Derek's comment in the chat about, you know, accessing academic resources, I guess, or review resources. Because I know that's something that you've talked about before when you were talking about like clear.
Okay, what is the statement in the chat?
Let me go ahead and I didn't want to take up more time necessarily pushing it here, but I'm curious. So when I do collaborations with people from other institutions professionally from the academic sphere, we have to do a cross-institutional ethics review prior to the work taking place, and this is kind of a barrier to having from the academic sphere reaching out to community projects because there is no venue for that ethics approval when there's no institution standing behind that. Have you thought of ways of to further incentivize people to participate, allowing this process to have some more of the legal trappings of review boards, so that they could allow these academic community projects to interleave more?
Yeah, okay. I think I understand where you're coming from. So last year and the year before, I gave some talks along with Tessa and Alex, Pearlman, Todd, along the lines of trying to form an ethics review board. And that's still very much at the forefront of what we want to do. So we thought, and the thought is definitely that let's develop, because there's funding for it, because iGEM is a collaborator, two is better than one. we can work on this biosafety and biosecurity board, and then we will learn a lot of lessons for how to set up an ethics review board for sure.
Okay, that actually helps hugely in how I think about this, in that I wasn't seeing the biosafety review piece as essentially being a training wheels for ethics, which is a great way of perfect, thanks.
Mm-hmm. Yeah, nothing else?
Any more feedbacks?
Yeah, Rachel, my open IRB dream for for sure. So I don't know how we are in time. 2:21 I've got. So I don't know what the organizers want to do, if we want to take a break and start our next section now.
So what's normally following is an update on some of the open COVID-19 projects, especially the diagnostic ones. So that will be a short session, about 20-30 minutes long. And then we'll have an update from the studies done by a group of academics at the Penn University about how the ethics was handled in the community environment of open COVID-19. And finally, there will be an open session, open discussion about how do we bring open community projects, bio projects that are fully open source and community driven onto the market. As this is actually something that several projects done by members of this community are struggling with. If we don't have any more questions, then maybe we can just take a very short break and let's see each other in this same room in 10 minutes. Well,
thanks so much.
Perfect. So first, thank you so much for everyone and your feedback. And please do get in touch with us if you want to also be involved and provide more feedback. We'll be also coming back to you also once we have more materials. We will want this whole process to be as transparent as possible. Otherwise, thank you again for Sarah and Tessa, and talk to you soon in 10 minutes.
Thanks so much, Thomas, and thanks all for your feedback. It's really appreciated.
Thank you.
Nice presentations.
Thanks, Rachel.
All right, so this is a second session, the second part of the JOGL session at the BioSummit, and we're going to hear from some of the project leads that participated into the Open COVID-19 initiative, especially specifically on diagnostics technologies. And we're going to hear first from Rachel Aronoff from Aquarium. And that she's been one of the main lead behind the Corona Detective project. And then we hear from Sarah Ware, and how do you call the project yet, Sarah? Because it's been through so many different names.
Our project is the 1 hour COVID test.
So the 1 hour COVID test. And then there will be opportunities for questions. All right, so Rachel, the floor is yours.
Okay, thanks a lot for the introduction. I will try and see if it will let me share the screen. We've been working more than a year and 8 months together for these JOGL projects, and I still can't believe how this pandemic time has passed. Can you see my screen?
Yes.
Okay, so yeah, the best thing I think about this pandemic was how we all got together and worked together on this. And the Corona Detective was really inspired by another project that had already been become quite familiar to Aquarium at the Open Public Lab, where I have been working as president for the last 3 years at least. And that's the GMO Detective project. And so I'll tell you a little bit more about that. But Guy Edelberg, who developed the GMO Detective at the CRE, he had done a workshop at the old Aquarium that I missed. And then he was at a citizen science thing in Geneva. And then for our 5th birthday party, he did a really nice Corona Detective workshop also. No, GMO Detective workshop for us. And so when the pandemic began, and I had for some reason, Toma was going to come visit us at the lab, and then we were all on lockdown, and then we were all pulled into JOGL, and we're like, okay, we're going to make a diagnostic test. Guy and I just said, okay, we're going to make a corona detective. And again, without the support of JOGL, probably our community lab wouldn't have been able to get the molecular biology reagents to do these experiments. And in terms of the biosafety discussion that we just finished, I think it's really key that we were able to, as a P1 lab, we've been inspected officially by the Swiss public safety people. We were able to acquire control sequences, both RNAs and inactivated virus, that really allowed us to do the research and development for this project. And it really was a great team working together. There was Fran Caro, Guy Edelberg, and I, who pulled together the Do It Together SARS-CoV-2, which became known as Corona Detective, for a LAMP in dark COVID times. And LAMP actually is the acronym for the molecular amplification method that's used for this diagnostic test that was also used for GMO detective. And so this is actually from Guy's thesis defense, because he got his PhD from the CRE during the time of this work. And it was all about open science and documenting our work and trying to figure out the best ways to do things side by side. So Fran at the beginning of the project was in Madrid, Guy was in Paris, and I'm near Lausanne in Switzerland. And as I said, the method was inspired by this GMO detective, but I'll tell you a little bit more about it. Actually, the method's been used for at least a decade to look for various pathogens. And so the GMO detective was all about looking for food products that had unlabeled genetically modified organisms within as the ingredients in the processed food usually. So especially corn, sweet corn, and soybeans are often genetically modified, usually to be resistant to the pesticide known as Roundup. And sometimes those grains do get into foods without people realizing it. And this is sort of that overview of doing a GMO detective workshop, but basically it's the same method for the corona detective, except we're starting with this RNA virus. So there's a very simple DNA extraction for the food products. For a GMO detective workshop, we have a very simple extraction for the samples, either saliva or swabs you can use for our test. And then the great thing about this method is that everything can be freeze-dried, and the frozen and dry reactions are stable until they're rehydrated. They can be sent anywhere around the world with no cold chain dependence. And so that's the main idea, was that we would be able to get these tests wherever they're needed without having to worry about this very expensive means of shipping things. And so after you rehydrate the reactions, then you add your DNA sample and you just keep it at the single temperature. Isothermal amplification means it's at one temperature for about an hour, and then with fluorescence, you can very readily detect what's a positive and what's a negative. In the GMO detective, they basically had a plant gene for the first 4 tubes and the GMO gene that was being detected in the second 4 tubes. But we've also been able with Corona Detective to do a sort of multiplex reaction, which I'll show you as well. So maybe I should have asked at the beginning, how many people would like a molecular biology sort of review, like what's DNA, what's RNA? In this slide deck, there are several slides we can go over. Maybe I should save it for questions in case I go over time. But basically, if you this isothermal amplification, again, I already said it means all at one temperature, the 63 degrees for 45 minutes. And the little teeny pellets here are basically all the reaction components. They have the nucleotides, the enzymes, magnesium, and a special sugar. And the special pieces of DNA, the primers, that specify our target in the viral gene. So here's a little graphic showing what the virus sort of looks like. It's little spike proteins on the outside, its RNA genome on the inside wrapped up in the nucleocapsid protein. This is how all the different genes of the virus are laid along its little RNA genome. And this little asterisk points to the part of the N gene that's our target for corona detective. And there's basically 6 sets of 6 different primers altogether that specify different parts of a short stretch, only about 300 nucleotides of the RNA. And that gives you the specificity that it will detect just this virus and not somebody else. And we did some work to make sure that new viral variants that arise during every infection, of course, would not cause problems in detection with our primers. And so from once these little tubes, you have them and you're ready to use them, you rehydrate them with a very small amount of the rehydration buffer, and you add your sample. And you can see that the method is super sensitive. So again, getting these controlled genes, both RNA and inactivated virus, that were P1 that we could work with in the lab, that weren't going to cause any problems for users in our community lab, was essential for us to be able to say that this R&D had worked at all. And I remember the first time I saw the positive with this beautiful green glow. And in fact, we can get down to a single copy of the RNA per microliter of the reaction. And this primer set that we're using has already been approved for 2 different EUAs in the States. And so that seemed like a good starting point, and we were glad to see how well they worked. But in those EUAs, they're not using the same detection strategy that we use. And it's called quasar detection. And the best thing about the quasar detection is that it gets rid of false positives, and it gives you such a strong signal, and you can look for more than one thing at a time. So actually, we've done already some different multiplexing with a cellular RNA and with other, like flu virus target genes. And basically, we know that this method is adaptable to anything with genetic information, from not just transgenic organisms that might be in your food or the virus that might be causing the next pandemic, but anything you're interested in. And it was really nice. The idea that of GMO detective from the start was that it was going to allow anybody to actually see DNA. And so again, if anybody wants to have more sort of molecular biology basics about how RNA and DNA bases are complementary to each other, I can talk more about that, but this is supposed to explain to you exactly how the detection strategy works. So what happens in these amplification, those little dry pellets have enzymes that make copies of DNA. And basically when a primer has its target sequence in the reaction from the sample that's added to it, then there's this large scale polymerization that goes on. First, the RNA virus has to get transformed back into DNA through one enzyme called reverse transcriptase, then the polymerase works. And it ends up making a very large multimer, many copies of the same sort of thing, but with this weird structure, because one of the primers actually includes sequences complementary to one strand and the opposite strand of the target area. So you get this sort of dumbbell shape, they call it. And in fact, once the molecule gets very large, it's more like a cauliflower, and they're very stable. And if you contaminate your lab with some of these amplicons, that can make it so you can't get rid of it, and you'll never get your negative control to be negative anymore. And so this is how, when we started in JOGL, Guy and I, we were just saying, be careful, don't open your tubes. I actually have a little bit of a delay, and I hope everything's coming through okay. But anyway, the bright tag that glows is fluorescein, and that makes the bright green color. And if you have a complementary quencher primer there, it actually has another molecule that sucks all the brightness away from the fluorescence. And when those are together and they're not incorporated in the amplicon, then that keeps it from glowing. So if there's no target there, you get, like in this case with saliva, you get no bright signal. And if you have your viral target there, then you get this very bright signal. And again, this fluorescence is very high signal because there's this sequence specificity to it. It solves the false positive problem that actually the other two methods that use these same primers had quite a problem with that, but we don't see false positives. And it lets you look for more than one thing at a time because you can combine colors. So say you have this one virus in green, the other virus in red, and both of them together you get yellow. But our most one thing we're really concerned about is that more surveillance screening is still probably quite necessary, especially in places where vaccines aren't being injected as well as we would like. Basically because each new infection gives the virus a chance to evolve and avoid being affected by whether or not you're vaccinated. So if, say, a new epsilon variant comes after Delta that no longer your vaccine isn't protecting you from, then we have a whole new round of pandemic on our hands. And so we're hoping that we can get our tubes made at large scale. And in fact, a Canadian company, EVIC, has made the first proof of principle batch, and we're hoping that we're going to get people to use this where it can be most needed. And here's some of the people from the great team, Guy, Tatiana, Fran, other people that we met just because of JOGL, like Ellen and Sarah, and Ferna and I already knew from the open science hardware thing, and also Toma Mboa. Of course, Scott is here. And thank you very much for your attention. And I see there are already some questions. I'm sorry, it's a bit late for me to start a talk like this, but let's see. What is the percentage of operationability? I'm sorry, I don't really understand what that question means. But it's,
so
hello.
How many, hi, very good presentation. I'm asking how many, if you're using on what scale and the percentage calibration, would you say the thing is effective? Is it 100%, 80%, 50%, 30%?
Right. So if we have, when we've done sort of with random samples from people reusing things that had been already tested with RT-PCR, which is sort of the gold standard, then our specificity is like 100%, but actually our sensitivity isn't as high as the RT-PCR. So I think it came out to something like around 80%. But if you compare to, rather than the RT-PCR, to the sort of antigen tests that people use, the quick antigen tests, but of course those you get an answer within 15 minutes. But for those, our corona detective is 1,000-fold more sensitive than those quick antigen tests. But we're a little bit less sensitive than the RT-PCR most in the random tests that have been done so far for clinical validation. It's a good question, thank you. are there hands up? Maybe I should stop the share, or was there? Oh yeah, there's. I saved this just in case anybody wanted to ask, learn about biology basics.
I think Rachel will have to go.
Yeah, we should stop.
To Sarah. Yeah,
I've gone too long.
Thank you so much for the presentation.
Yes. Thank you.
I have to unmute once again. Okay, so let me try again to share the screen. Okay, so our test is called the 1 hour COVID test. And when I say our test, it's more than the people on this slide, really. As Rachel was pointing out, we have been working as a bigger team on collaboration and sharing information and suggestions among teams of different tests, let's say, that are towards the same goal. And so Rachel already gave a nice presentation on the technical aspects of LAMP. And so all of our tests are involving LAMP detection. So it's the same idea in general. But so more particularly, the people that were doing the lab work and on this project were Ellen Jorgensen. She's Chief Science Officer now at Annika Biosciences, and she is also with Biotech Without Borders. And of course, formerly GenSpace. I love this picture of Ellen. I need some kind of iconic science photo like this. Chris Monaco, who is a microbiologist with the CDC in Atlanta and formerly with Decatur Makers. And then myself with BioBlaze Community Biolab in West Chicago, Illinois, and also a sister lab called Blossom Biolabs. So Blossom Biolabs in combination also with BioBlaze Community Biolab, but we have BSL2 capabilities. So certainly we have the biosafety cabinet and things that are certainly useful for human diagnostic testing. So this is an overview of our test. So we start with a collection kit that is FDA approved. This collection kit is an oral swab kit. And so you just swab inside of your mouth, kind of all areas except for trying to avoid the teeth. This comes with a buffer. So you go ahead and swab your mouth, and then you unscrew that swab and you put it in the opposite direction. It goes into some buffer that will inactivate any virus present. It will lyse any cells present, and it will stabilize the DNA, but for our case, the viral RNA, so that it doesn't degrade. So this was something very important for our group, our team in the beginning, was that we want something that will inactivate any virus if it's present right away. And we can store these at room temperature until testing. So then there is a very simple RNA processing step, which involves something called PrepIt. So there's a reagents kit called PrepIt, and this is just adding 10 microliters of one thing to 100 microliters of sample, and then adding 20 microliters of the next thing. And you pipette up and down 12 times, and you just let them sit on the bench at room temperature for 15 minutes. And this is a phase separator. And so all the junk and the cellular debris will settle to the bottom of the tube, while the nucleic acid floats to the top of the tube. So then there's a dilution step, because we need to dilute out some of those enzymes that are stabilizing that RNA, because we need to then use enzymes in our reaction. So then we just put these samples in my lab. We use a sous vide precision cooker. So it's like $100, and it keeps a more constant temperature by far than my traditional lab water bath. So this was a suggestion by Guy Edelberg, thanks Guy, that's a great suggestion. So this was is our water bath. You just need one temperature, as Rachel was mentioning. We use 68 degrees Celsius, and then you see results that are based on a color change because of a pH shift if the virus is present. So from sample collection to results, this is within 60 minutes. So it's not a rapid test, so it's longer than 15 minutes, but it's not a PCR test, which is 2 to 3 hours from start to finish. Actually much longer than that a lot of times, but anyway, and we're somewhere in between with like what Rachel was saying, not as sensitive as the PCR, qPCR test, but certainly more sensitive than the rapid test. So we don't need any centrifuge. We don't need a thermal cycler. It's just you need very minimal equipment. You need pipettes and you need a sous vide precision cooker. Okay, I'm trying to advance my slide. Okay. So Rachel didn't mention, but I'll give her a shout out as well. But the 1 hour COVID test and also 3 other groups in JOGL, including Rachel's group with their corona detective, we made it to the semifinals of the Ansari X Prize for rapid COVID testing, which we thought was phenomenal, not just for personal reasons, but for community reasons. So we had 4 open source teams make it to the semifinals for this X Prize. So, congrats and kudos to Open Science. So this is something that we needed to do for the X Prize. And so you can see 20 reps of positive sample, and these are contrived samples. So meaning samples that we added inactivated virus to the tubes. So these weren't patient samples, these are contrived, but we were getting, detecting about 200 copies per microliter of the inactivated SARS-CoV-2 viral particles. And then you can see on the right side, the pink means negative. So these did not change. They were only water controls, no virus added. And then you can see the human actin controls. So we also had included human cells. And so these should be positive no matter if your unknowns are positive or negative. So this was with contrived samples. We did have some initial, we have more now, but when this photo was taken, we had 9 clinical samples. So these were people who came to the lab and they were tested, and we had some of these that were confirmed with RT-qPCR. So this first set of 1 through 6, these were confirmed with RT-qPCR. The patients, the people went ahead and they'd had our test done, but also the RT-qPCR standard test. These were all negative with both. And then we had a confirmed positive. Well, let's talk about this one first. So then we had some that were confirmed with the rapid antigen test. So these results matched with ours. One was negative, one was positive. But then we have number 9, which in our lab, I detected as positive. This person then went and had an antigen test done, and that was negative. And we thought, uh-oh, this is not matching up. But then the same person went on to get an RT-qPCR test done, and it did come back as positive. So we were able to detect something that the antigen test was not able to detect. And then we have our positive and negative controls here. We are now in the beginnings of clinical trials with patient samples for the 1 hour COVID test. So time will tell. We will see what that yields. And I don't know how we are on time. That's all I kind of wanted to say for our update. But so Tomas, maybe you can tell me, or Xavier, but Rachel, I wanted to have us both talk a little bit either now or during the session after the UPenn results are given, talking about lessons learned and how we have approached it differently for how to get these things to the masses of people and our struggles in that. So Tomas and Xavier, would that be for now or for later?
I think we could actually take it for the JOGL Fitz, as it's very much related to
Okay,
to the
let me
scale up.
stop sharing my screen.
Like this, we're perfectly in time for the next talk.
Are there any questions for me, or do we have time for that?
Yeah, we have time for 1 or 2 questions, definitely.
And following will be a break that will run until 4:15. So I imagine they could run into if y'all wish.
So, okay, no questions?
You can still write your questions in the chat.
Additionally, everyone can raise their virtual hand as well.
Okay, well, thank you for listening.
Thank you, Sarah. Thank you, Tomas.
Should we ask a question? Does anyone want to get tested for COVID anymore?
Well, I mean, like to that point, you know, as Rachel pointed out, well, certainly in the US, people are still getting tested. There's the elementary school where my kids went until last year. One of my kids finally they're out of elementary school, but their former elementary school in Chicago area, the suburbs, they went to e-learning for all of last week again because of COVID cases. So we're actually spiking up in our area around Chicago again. But to Rachel's point earlier, I mean, one of the great things about developing these tests is that you can substitute primers and you can use the same pipelines to detect other kinds of pathogens, not just viral, also bacterial, fungal.
Thank you, Sarah. Thank you, Rachel. All right, so during this time, we have a break that'll run from now until 4:15. Everyone can take some time to walk about, do what they got to, or also allow some discussion to continue to flow over as needed. Thank you all. Everyone, please thank our speakers.
Sarah. And yes, definitely Sarah and Rachel. So the next session is going to be novel biosafety and bioethics questions in the JOGL Open COVID-19 program starting in 15 minutes. And so we can just hang out in this room until then, and we can discuss if you would like.
Maybe we should talk about serendipity and how things just happen. It really was funny, I thought, because we had talked on the phone in the February before the first lockdown, and you were supposed to come to Switzerland, Thomas, and then, you know, it just happened.
It's true. It's true. Yeah. Yeah, I was supposed to actually come to Sweden for the inauguration of Innovation Space. I forgot the name of it now. And I still remember, like, they were not considering the COVID-19 at all. You know, it's like so at some point I wrote to them and to all the speakers saying, hey, I'm not going to be able to come due to the situation. And the responsible of the Innovation Hub wrote me back, say, how can you do that? Do you want actually to threaten the events? Like, I mean, just look at the numbers. It's going to be canceled anyways. And then she replied back saying like, yes. She thanked me later, but yes, at the moment, like people were just lost and didn't understand what to do.
Yeah. And that's still my big worry, is that people keep saying they want to learn to live with the virus and you know, let it spread around, and there's just still too many people that have no immunity and are ready to pass it on, and we're going to have new variants. And who knows where the one that gets us next will come from.
Mm-hmm.
If I could share a thought.
Yes.
I am actually wondering, like, how long this road still is. It's super long. I mean, you guys did an amazing job with the production and the quality control and organizing everything. I mean, I see how much work it is, Rachel. But when I'm thinking about this talk I had at Reclone with the medical guy, and it was about like real world implementation about in rural areas that really needs this.
Mm-hmm.
And it totally like flipped my mind, like, yeah, but if you have this detection method and you have no possibility of quarantining the people or treating the people, you know, it's also senseless. You know what I mean? It's like the whole picture is actually needed. It's still so much.
Like refugee camps and things like that. Is that what you're thinking about too?
Yeah, for example, like the real rural areas where you really have like a lot of people needing help. And yeah, the more infrastructure fails, the more work you have to do until you can really like help there and not be like just another project that wants to verify stuff, you know. And yeah, there's still somehow a big gap, or at least in my mind.
I mean, hindsight is always, it's 2020, they say, but I still don't know if it would have been better if, like, say we had someone really like an entrepreneur that made the company. Maybe it would be used right now, but would we be any better off? I don't know.
I actually, but it's just my personal experience. Like, I like the way it develops slowly. And there's so much space for exploration if this solution ethically really fits, you know. And with scaling up too quickly, it's, of course, you need scaling at some point, otherwise it also disappears. But if you scale up like too quickly, like, we have this idea, we have the prototype, now we scale, and you don't have these iterations you guys have with all the calls and all the work, then yeah, I guess it's more like the startup problem I see in Germany, that you have so many solutions, but actually no market, because nobody thought about, I don't know, how will you use it in some kind of context, right? So I think the way Corona Detective develops, it really tries to fit.
Well, we still have, I mean, too many different use case ideas, but nothing is really out there yet. So we'll see.
Yeah. It's exciting to follow this project, really.
Okay, so what is this? Well, we're going to talk a little bit about our project. And first, Lisa and I will introduce ourselves, and then we're going to talk through sort of what we did over the past 18 months and what our findings were in regards to biosafety and bioethics questions on JOGL through the Open COVID-19 initiative. So I think that this is a step up, step back moment for me. I have been talking for 3 days, so I think you know who I am. I am an organizer of BioSummit. I work at MIT at the Community Biotech Initiative, and also I'm a researcher at in the Wexler Lab at University of Pennsylvania. But Lisa, please tell these lovely people who you are.
Hi, everyone. I'm so thrilled to be here. I've heard about BioSummit for a while, and I hope at some point to come in person. I'm a professor in the Department of Philosophy at UNC Charlotte in North Carolina, and I've been teaching research ethics for quite a while. And as I started to do that, I started to find all these areas like citizen science and DIY bio and biohacking, where the usual approaches to research ethics, including regulatory approaches, don't work. So I've become really interested in what are the ethical stories to tell, and can I be of help? What can I learn? And so that's how I found Alex, and Alex found me, and I found some of our other collaborators, and they found me. So it's been a serendipitous partnership.
Like everything in community bio, right? Okay, so Lisa has a phrase that she coined called the ethics gap. And it's something that we talk about a lot, where there is this sort of space where community bio projects exist. And at this point now, some of them are becoming rapidly very mature and ready for prime time, but there's very little oversight. There are no infrastructures that sort of are built around overseeing the ethics of community bio the way that there are for institutionalized and establishment research. And so we wanted to sort of look at what was going on JOGL in this ethics gap. And so we attempted to come up with what we thought as sort of like establishment researchers and what had been in the literature, what the fears were about where that ethics gap sort of existed. And we picked out some places, and specifically those were biosafety and harm, validation and replication, and authorship and credit. And so using those 3 buckets, we went into JOGL, and with help from Tama and his team, and people like Sarah Ware, who's here, and Todd, who are consultants to our project, we were able to gather a ton of data about JOGL based on keywords related to these 3 buckets, and went through all of the Slacks. So everyone who's been on JOGL, we've been talking about these issues. We have been keeping this big eye on JOGL, and like we're trying to report back on like what we're finding with some methodology. And what we actually found was that like those are not the buckets we should have been looking at. And it turned out that actually the ethics issues and biosafety issues that were existing on JOGL weren't exactly the ones that sort of the traditional literature have picked up as being important. And so we'll go into sort of why we need a paradigm shift to look more closely at the issues that we did find. Okay, so methods. Basically what we did was we had 2 sort of courses of our methodology. So the first one was a digital ethnography, and what we really mean by that is we, like I said, went through JOGL with a fine-tooth comb based on keywords, and we downloaded the Slack archive with the help of the JOGL team and went through it. And with thousands and thousands of pages, we read them all and highlighted and picked out places where there were conversations related to the 3 buckets that I had mentioned. And so we had 50+ search terms ultimately, We looked at 107 projects and thousands of users and thousands of pages. And so from that, then that informed sort of the next step, which was interviews. And so we, together as a team, created a series of interview guides for 3 different groups that we identified as sort of like the JOGL stakeholders that we wanted to really focus on. And so the first group was a biosafety group. So people who were involved in writing the JOGL biosafety guidelines, people who were active on the JOGL biosafety Slack channel, and people that we knew had advised those groups through conversations that we saw on Slack. The second group was project leaders. So 107 of the projects on JOGL that were listed under the COVID-19 initiative did have sort of leaders listed. And so we went through in just a big spreadsheet and found the ones that were using biologics or maybe had biosafety or bioethics questions. And we emailed them and asked to interview them. And we had an interview guide for that. And then finally, we did talk to JOGL leadership. We spoke to a number of people who had been leaders, not just at JOGL, but also on the Slack during the sort of crisis time for early COVID, and who had been organizers and sort of like guideposts on the Slack for the Open COVID-19 initiative. And then we underwent 2 levels of sort of coding and analysis to go through all of these papers. So I'll just really quickly talk about some of the results. So we sort of found 2 levels of results. One is biosafety and one is bioethics. And I'll just focus on biosafety quickly, and then I'll turn it over to Lisa to talk about our findings on bioethics. But basically, the biggest takeaway was that the culture of biosafety on JOGL, which existed and is very deep, that we found, did not come out of nowhere. Like, I know we all heard from Jenny Malloy this morning. She's amazing. And we also heard from Thomas. And for the past couple of hours, I know you have been talking about Fitz and other issues. It's very clear from just even spending a little bit of time with people on JOGL and around JOGL that the leadership of this community has been engaged in a biosafety initiatives and interested in that for a decade. And the people who have been working together, knowing each other, this global network, when there was a crisis and there were no biosafety guidelines, leaders were organically already in place and able to work with each other because they knew and trusted each other to create a sort of net around JOGL. And it was like this very magical right place, right time situation. And sort of through that lens, what we found was because the biosafety guidelines were already drafted in the handbook that we brought out at BioSummit last year in this track, like I said, people had trust with each other. So we also found that the role of the Biosafety Board on JOGL, which was a group of, I think it was 9 or so. I think some of you are here, so you can correct me if that's the wrong number, but I believe it was 9 people on JOGL who stepped up into the role of being this Biosafety Advisory Board in case anyone on JOGL had any questions about their projects. These people and the JOGL leadership were also, because everything was sort of in flux and it's a building the plane while you're flying it situation, like so many things in community bio, there was not agreement. I don't know if I would say disagreement, but there were different opinions about whether that board should be a regulatory, like approval making body, or whether it should just be advisory. And I think that that conversation is still in flux. But it was a really interesting to sort of see where and why people landed on that issue. And then finally, I think the peer review system that we heard Thomas talk about in detail this morning during his plenary matches so well with like this innovative biosafety advisory group that sort of jumped out, because most of the projects on JOGL reported that they did actually know about the biosafety guidelines because they needed to check off like, yes, I agree to be safe in biosafety when they applied for grants through the peer review process. So interweaving those 2 things, ended up resulting in more people on JOGL knowing and being reminded about biosafety, and that there were resources available to them should they have questions. So that was really, really integral. Okay, so I will hand it over to Lisa.
Thanks, Alex. And I'm not sure how long we have, so if somebody wants to just stick a couple minute countdown in the chat, that would help. So one of the places we were coming from was thinking about this in what we call an establishment framework, which means something like the traditional research environment and system and set of structures that you are familiar with in terms of having research mostly located at institutions of higher learning around the world that give degrees, they train the new generation, et cetera. And so when we came to this project, we had those couple of different buckets of ethical considerations in mind, but they really came from the establishment world. And we knew that we wouldn't necessarily only find things from the establishment world. And in fact, that's what happened. But we wanted to find out what people were actually seeing as ethical problems in the context of JOGL, in the context of the pandemic. And so we did find a number of issues that'll be on the next slide. And what we're arguing, and Alex is leading the paper on this, we're going to argue that we need to make a paradigm shift in what counts as research ethics. And for those of you who don't know, in the United States, at least, the National Science Foundation and the National Institutes of Health have standards for what are typically counted research ethics issues. And so even though they don't mandate all and only those areas, it turns out that people trying to figure out what to teach in a research ethics class just look at those issues, and they teach conflicts of interest in human subjects and animal subjects, right? And they're just categories that don't fit DIY bio, community bio, citizen science. And so Alex, if you want to shift to the next slide, we'll just give you a few examples of these. So here are some general categories of ethical, what people saw as ethical issues that don't normally count in establishment ethics as research ethics. People will say they're ethical issues, sure, but they're not research ethics. So for example, peer review and funding. Part of the question is both a quality consideration. I'm sorry, my dog is right next to me and he's going to pick now to get a little upset, so apologize. So peer review and in particular funding was a worry because a lot of people on JOGL really needed some funding to help them get going. And one of the things that became clear as we were looking at the data is these micro grants were everything for a lot of the projects and enabled them to just actually get going. There was a lot more conversation about intellectual property than I think we had anticipated, not in the sense of the projects claiming intellectual property, rather that they were fighting against existing intellectual property in the world. And this is an ethical issue if you think about it in the context of a pandemic. So we're trying to move fast, like Sarah's trying to get a testing regimen going, right? And you have to pay for something that's under intellectual property protection. So that really can impede things because you have to find a workaround. And there was a huge consensus about access to resources being an issue. People would come into the platform and not know where to go, and they'd have to try and find each other, and they'd have to onboard, and they were trying to find someone with the expertise to do X and Y and Z. So that was a barrier to them moving faster than they otherwise might have. And when we say here the leadership and structure of JOGL was an ethical issue, we don't mean that there were any problems with the ethical nature of the leadership and structure, rather that because it was thrown together so quickly and had so many people jump on so quickly, that the structure and trying to talk to leaders and having leaders have enough human time in the world to answer questions, to help people find each other, to build the structures that were necessary, just couldn't happen. And then finally, the equity and social justice was, I think there were a lot of little threads in there. And Alex, jump in if you can remember some of them too, but I know there were some in particular in other countries where people were trying to find expertise even at the same time zone that they were trying to get money for resources where they were trying to make sure testing happened. And a lot of the motivation for the platform during the pandemic was, let's get the things that people need to under-resourced countries as soon as possible. So I'm going to turn it back to you, Alex.
Yeah.
And see what else.
And I know Thomas wants to wrap up, but I also just wanted to add through the thread of like this Bio Summit that I've heard over and over and over again is that time is a resource. Time is a resource, and like knowledge that's outside of your own expertise is a resource. And like those are things that are missing just sort of across community bio. And it was something that we heard a lot about in conversations with project leaders on JOGL. So I'll just wrap up with just sort of a overview of what we found, and what we've been saying was that there's already this culture of ethics and biosafety. And it's becoming very annoying anytime a new academic sort of enters the space or the news media and is like, oh no, the biohackers, they're so unsafe, what do we do? And it's like, no, well, we need to keep like talking about that this culture exists and has existed for a very long time. That said, the ethics and infrastructure tools just look different, and they're going to be different. So what we need to do is start creating tools and infrastructure to approach different problems than maybe exist in traditional spaces. So additionally, yes, this situation was magical because it was a right place, right time, where everyone who was building the plane happened to know each other and happened to like be able to already work well together. And that was fantastic. And really helped the collaboration move smoothly. And finally, that the motives were pure. Everyone who came to JOGL to work on these projects wanted to do something to help in the effort to combat the pandemic. It was very obvious that everyone there was going with good intentions and assuming that everyone who was also on JOGL were going with good intentions. And I'll just say, and I don't think we're writing this, but we also didn't see a lot of fighting. Like usually when there's like threads and message boards and stuff, like people are at each other. I didn't see that. And I think that's like, that's just the heart emoji for me. Like that was the core of just the beauty of JOGL. So that's the end of our talk. But also, Lisa, here's a plug for please submit your abstracts. If you have papers, Thomas, I know you're writing papers. This is a special collection that Lisa is one of the editors for Citizen Science Theory and Practice, and it's a special collection on biomedical citizen science. So I think that probably everyone in this room has something to say about that. And so please submit your abstracts for this special issue. And thank you so much for listening.
Wow.
Sorry, that was so fast.
No, it's amazing work. It's fascinating actually to have an outside look at what we've been living every day from the inside. So yeah, I would love actually to see more of the details. Like, how do you plan on sharing these results to the public?
Yeah, so a couple of things. So first of all, Bio Summit happened before we put together our one-pager for you that we were like, Thomas, we have results. How can we do it? So that's the first part. So we do want to host like some sort of like meeting with you, the team, and then figure out how to do something maybe bigger with more collaborators and more folks on JOGL. And yeah, we're just writing a bunch of papers because also we are like traditional academics, and this is an NSF funded project. And if we don't write papers, then I don't know what they bring us up. What happens? I don't know, but we have to do it.
But we do write papers too at JOGL, because it's the best way actually for this kind of research to be cited by the more traditional academics too. Yeah.
Right, but like, why do we want to be cited by like what? But is it the best way? And why don't we do interactive websites or like build an app that just like quickly explains what
I know. We'd get there. We'd get there.
Yeah, we're gonna do both. So we're gonna be very creative. We're gonna be very creative about it. So yeah, and hope to work with you and the rest of the team at JOGL. And yeah, figure out what's the best way. I don't know if anyone has ideas, like if you're here and you have a way that you want to access this, like please let us know. You know, you can find me, you can find Lisa, and Sarah is here too. Anyone who's on our team, like please let us know, like how would you like to access this data? Yeah. I unfortunately have to jump back into communicating science where I'm I'm sorry.
But it's been amazing. Yeah, thank you so much. Amazing work, and I guess a lot of people here would be actually very interested to dig more into the results and the data. But yeah, it's been a pleasure collaborating with you. Thank you so much for
you too. Okay, well, I'm going to sign off. And Lisa, thank you so much for being here. First Bio Summit. I hope you hang out and go to the other rooms too. Like, come to Communicating Science. Like, let's all hang. I'll see everyone later at the plenary. Lisa Joy at 7 p.m. Okay.
All right. So thank you so much again, Lisa. We're going to go to the next session and final one for the JOGL special session. And this is one about JOGL Fitz. JOGL Fitz is really an experimental idea which has no shape yet. It's a discussion that we've been having with several members of the JOGL community, specifically people who are deeply involved with the Open COVID-19 initiative on the diagnostic side of technologies. And the idea is like, how can we support open bioinnovation that have been driven by communities, and especially like members of this community, where these members don't want to change life. They don't want necessarily to become entrepreneurs. They would better stick to be able to contribute continuously to the improvement of the technology or its diversification. It seems that a lot of those ideas cannot just take the traditional form, the traditional shape of a startup and go into the startup path. Like, how can we actually make sure that the beautiful work done by the many communities contributing to the commons can really be made available to the public. And so this was introduced actually already by Martin yesterday in a talk. And so I'm just going to reuse the slides, but it's mostly actually an open discussion. So just a minute when I actually go back to the slide. Oops, yeah, here it is. So those are the slides that Martin used yesterday. This is going to make it easier. Yeah, so you heard in one of the previous talks that some of the JOGL team made it to the semi-final round of the XPRIZE rapid COVID testing. In fact, even one of them made it into the final, and they were the only project that were community driven, and so we're quite proud of that, and it's a beautiful story that needs to be spread. And so it's as I was explaining, that is how can we build a pipeline that can truly help those open source project, community driven projects get into the market. So it's probably actually Martin would be great at explaining this, because these are his slides. Martin, are you here?
Yeah, I am actually. Yeah.
Yes. Actually, I think it would be better, since you've practiced yesterday, would you like to go through those few slides that you made? I think it would be easier.
Sure, yeah. Practice is always good with these things.
Yeah.
So where are we?
Do you need me to go back?
Yeah. So basically everyone saw a huge amount of, even in the media, the amount of groups that came forward and wanted to work on various different projects, totally because they just wanted to solve the problem during the pandemic. And it was actually, for me, it was quite exciting. It was very exciting actually to get up every day and not have to be signing contracts and non-disclosure agreements and dealing with patents and all this stuff. And I think that teams that did work in this area made progress really almost exponentially faster than it was possible to do if everyone was fighting against each other and keeping all of their capabilities secret. So it was really, really liberating. Now, that came with the kind of counterpoint that very few of these projects ended up in an actual product making it into the market. And by market, I mean actually helping a patient in some medical environment, especially in a production environment, let's call it, rather than just a clinical trial environment. But I think a clinical trial environment would have been really valid as well. So next page, please. Yeah, so why was this, or is this? I think it's because of the unique challenges of operating in this highly regulated environment. A lot of scientists that, just want to help, a lot of engineers that are out there that know how to do it all. And the scientists know the biology, they know how to make it work, but they're not aware of the regulatory challenges. And also the general challenge with regards to translation, right? You need to get access to clinical data. You need to have access to medical practitioners who are willing to trial your intervention or diagnostic or test it on human patient samples. Even getting access to patient samples proved incredibly hard. A lot of red tape that tied a lot of stuff up for a long time. And yet you can take a sample of your own skin and give some a data set or saliva almost instantly, right? And yet to get anyone else's, the amount of hoops everyone had to jump through. So there's some of the challenges. Then again, when you say and you contact clinicians or medical institutions or regulatory authorities and say, hey, we want to give you this, they're like, what? You want to give us something for free? Like, yeah, and it's amazing that it works. They say, no, they don't get it. Unless you're approaching them as a formal entity who has all of the backing and all of the kind of institutional might of, not might necessarily, of a traditional corporate type entity with the right names, even like CEO, blah, 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 CTO, Chief Science Officer. It meant that projects weren't represented, open projects weren't represented in the right way to clinicians and medical practitioners. And that kind of means that a lot of these things are destined to remain at the proof of concept stage. Okay, next. So yeah, so that brought us to discussing the creation of an entity that we ended up calling Fitz, which is named after Alexander Fitzgerald, a Canadian, who was instrumental in some of the insulin stories and in some other typhoid, I think it was, projects. So we kind of went, we need an entity that can actually address all the things that I mentioned on the last page, from the open scientists, the engineers, the scientists, and the medics who are in our community, and represent us to be the interface to the traditional medical institutions, the traditional regulatory authorities, etc. So we call it Fitz, and we've been chatting about this now for a while, and we want to chaperone projects that have the capability or the possibility or the desire to move into the clinic and become a product. But it's very important, I think, that we want to be open source. We don't want to create a company for the sake of a company. This is not about making money per se. It's about getting a project or a product into the market that wouldn't get there otherwise. And we want it to be open so other people can build on it, just like Linux. We want people to fork this. We want people to fork it and make money for it themselves if they think they can. We just want to get the projects into the market in whatever way possible. We also would like to run an open institutional review board, which was kind of alluded to a bit earlier. I'm working a little bit with that on set with Sarah Ware on that. We want to counsel these projects and in some cases provide funding to them. So and also public outreach. We want to, you know, be a face and advise projects on how they should represent themselves to the public. Okay. Next. The next page, I think, is just kind of a visual representation of what I've just explained. Okay, next. So this is really important to me anyway. We want to keep it all open source. I think everyone in the community generally wants to work that way. And it's very important that you know your legal issues, because when you're dealing with people who aren't aware of the reagents they're dealing with, they're not aware of the fact that a reagent might have loads of patents on it, especially when you go commercial with it. In a lab, it might be okay. And you might get it to work. But once you go to, I'm not even saying commercial, once you go to a regulatory authority and say, we're doing this in anger, so to speak, so in actual patients, there might be changes in your licensing with regard to that. So it's important to know that and have everyone discuss that at the beginning, because you could elect to go down one road, one readout, and that readout could be fully open source all the way, but another readout could be patented. And we actually had an issue with that in that one of the readout texts was protected in one regime and not protected in another regime. So academics may not be fully free, I think, as well, to help in some of these areas, because in a lot of cases, the universities will say, well, that's our work, even though they might be doing it off hours. So that's just something people need to be aware of. And again, you know, we want people to fork these ideas and fork the projects and fork the technology and even fork the company or fork whatever gets formed. We just want to see open scientists create projects and products that make it to the market and that can assist in increasing the level of patient outcomes that mankind, I think, is capable of doing, but currently isn't. I think that's about it. Yeah, of course, these were our Pathfinder candidates: Corona Detective, Viral Alert, and of course the 1 hour COVID test from Sarah is also in there. I think that's about it, Thomas. Yeah, this is kind of a timeline frame, but I think that's really functionally about it.
Maybe this one could be of interest.
Yeah. Do you want to talk about this, Thomas?
Yeah, I can. So we've been trying to think of a way to, you know, how do you make sure that a community who is dedicated to the advancement of a technology, of an open source technology, has the ability to continue doing so, while the results of this research and development can be used by commercial partners. And so to create a link, we need to create a link, an interface between those commercial partners, those industrial partners, and the contributing community. And so that's where kind of like where JOGL fits in. And so you know, it could work through a set of agreements where JOGL fields would actually represent the community and as a formal entity, and would be able to negotiate, for example, if the commercial entities would like to do so. So as Martin said, that there is no obligation. The company could just actually take whatever is are already produced. But we believe that being able to if a company is interested in technology, they probably want also to be able to benefit from the inventors, from the contributors. And so so because they will not be able actually to contract anything directly with the community, they would contract with JOGL Fitz, and JOGL Fitz would just direct whatever assets, funds, you know, or data, you know, that would be provided to the community, and would guarantee that. Yeah, and so this is kind of like new. We are not aware of anything that is close to this kind of functioning. Would love actually to hear from you if you had actually thought about that before, if you've seen something similar, if you are facing similar issues. This is very much an open discussion. This is something that we are all interested in this group in creating. We are aware that this is very complex. It will require also a lot of legal expertise, and this is going to be a long road, but a definitely worth one on the far end. I guess this is it, and we could just open discussions.
This is Lisa. I'm really excited to see what you all are doing with Fitz and to see how it's developing. And I've sort of heard stories about it percolating in the background from conversations with Sarah and Alex. And so it's nice to see this sort of big vision, which includes the incubator and the IRB and, licensing and open source and forking and everything. So my mind is mostly kind of hovering around the IRB aspect of it. That's what I'm most familiar with. And I'm also wondering, how are you thinking about international differences in terms of how they handle IRB oversight and what they require in terms of IRB oversight before you take things to market? I think, let me just add one other piece, that there's a lot of projects going on in citizen science as well that aren't maybe driven to produce the kind of technologies and services and products you're talking about, but that do involve human subjects and would be interested in trying to take advantage of an open source IRB, since many of them can't access them. Cost is an issue, but I'm trying to think about how the first barrier I see is this international question.
It's something that we have actually faced already, because we've been trying to interact with some of the European institutions, like the regulatory institutions, and Martin was one to initiate such an interaction in Ireland, where he's based, until actually we understood that even in Europe, you have actually to deal with institutions that are from within your own country. So as JOGL is HQ'd in Paris, we need to go through the French entities. So this is definitely an issue. And however, if we create, for example, a pipeline with the French institutions, then automatically it would open up to the European level. And so we create such a pipeline at the European level, but that wouldn't work for example, the FDA in the US. And so that would mean that in parallel, a team will need to actually work specifically on the FDA pipeline, for example. And so we've been talking about that. It's one of the reasons why we actually created JOGL North America, 501c3 based in New York, in order to be able to interact with American institutions. But we are at the very beginning of this process.
Thomas, do you want to, I don't know, maybe you don't want to mention, do you want to mention the US entity?
I just did.
Oh, that was sorry. Okay, yeah. So we're trying to do that. Yeah, for sure. We're trying.
You want to give more information about the Open IRB, Sarah?
Open IRB, yes. I mean, it's the thing that we've been talking about for 2-3 years now, right? And the idea that we need some kind of an IRB for the community, because of costs, just because, of access, and we had this idea floating around for a while, but it was like where to house it.
Actually, Sarah, could you just reintroduce for people who doesn't know what is an IRB?
Sure. So it's an institutional review board, which is an ethics review board. So why do we need it? Let's start there again. So we need this because in order to get health related products through regulatory, through clinical trialing, that's like step one, is that you need institutional review board approval. So an ethics approval, so that your project has gone through some scrutiny and that people are protected. So it helps to protect marginalized people, and it helps ensure that things are done ethically. So we were talking a little bit earlier in the session today. The first talk of our JOGL session was about creating this biosafety and biosecurity review board, which is something different than an ethics review board. So both of these are very important for our community and for any type of research involving human subjects. So, so where to house this? And even I had thought about, because I have a nonprofit community lab, I also have another lab that's I consider a sister lab to BioBlaze, called Blossom Biolabs. But thought about trying to maybe house this within one of my labs. But in the end, we exactly what we just talked about, what Martin was mentioning, is that we need, it's not about doing something sneaky or something like that, but it's about giving regulators what they're used to. What they're used to is not, hey, my lab is in my garage, or God bless you, Rachel, or my lab is my shed. They're not used to that. They're never going to be used to that. I'm sorry, they're just not. And so what can we do that's not compromising our beliefs and our community values, but still looks like something that regulators are used to? And so this was our premise and our idea for JOGL Fitz, one of the premises, but was that, you know, let's create this entity so that there's something that's a global research institution. Well, that's something that regulators are used to. That's palatable. And so within that, of course, we would have an institutional review board within our research institution. That's the normal thing. And so it's an idea to make this work. And I know in the past, there's been a little bit of pushback, not a ton, which is nice and a little bit surprising, but there's been a little bit of pushback as far as like, why should we try to fit into the system that we are trying to not be a part of, or that doesn't want us to be a part of the system? And why can't we need to create a new system? And I'm like all for that. Hey, by all means, try to create a new system. But right now we have the one system to work with. And so, you know, let's maybe think about doing things in parallel. And while we are trying to create a new system, let's try to work within the one we've got.
So Catherine is asking a question. Can you tell us more about how you differ from a local innovation hub? I can try to provide an answer to that. In my knowledge, what would local innovation hub do is actually mostly focus on startups, companies, company entities, and they would actually not necessarily consider working with distributed communities. I don't actually know any local innovation hub doing that, but I may actually not have the same definition of a local innovation hub as you, Catherine. So if that's the case, I would love actually to hear from you if you have an example of such entity doing that. But the thing with innovation is that it's mostly actually considered only at the company level. And if you're not a company, it becomes very hard actually for people to consider your effort. So that's why we're trying to create this hack where JOGL Fitz will become a representative and will hide the fact that behind it's actually a distributed community, a decentralized community advancing the research, and not a structured entity that is fully registered.
Yeah, and Thomas, I'll also add to that in that anytime I've spoken to local entities that are looking to kind of fund you in a small way, or even in a big way, they're all looking for IP protection. They're saying, what's your moat? And you're like, well, we're not actually trying to create an IP moat here. We're not trying to stop people from progressing this science. We're trying to prove the science works, get it out there, and allow other people to make it better. I think that's a big thing that most traditional funders would be like, we don't really get that. And there's ways to monetize that, as Vikram says just below, you know, that there's lots of avenues. Once you're seen as the people who are pushing forward the biology or the science or the software in the case that he mentioned, opportunities arise to monetize around that. It could be just your brand. Something comes out of JOGL, it's considered, the brand alone is very valuable. Or sorry, Fitz, JOGL Fitz. So I think there's figuring out to do there, but I think that if you create something valuable for patients and you don't have massive costs, you're not having to pay a very, very high salaries to all of your researchers, because most of the people involved will be working relatively cheaply. You can be cost effective in getting your product into the market and making a return for building the system, building the flywheel.
So while we're working towards this JOGL Fitz, we're really hashing it out and learning a lot of things as we go. But right now we don't have it in place, ready to go. So I thought it could be, or we thought it could be interesting for Rachel and I maybe to mention what are the things that we are trying to do right now in the absence of a JOGL Fitz, in the absence of a pipeline to get our diagnostic tests out to the masses. Lessons learned, we like to say. So I don't know, like Rachel, you want to talk about how you guys are trying to get Corona Detective out to the masses, and what are some issues you guys are facing?
Yeah, so for Corona Detective, it's really been an extra challenge because we really wanted to focus on places that are not well covered by vaccines, and places that we already had some collaborators. So there were people in Chile, there were people in Sri Lanka, there were people who helped us with the X Prize semifinals part. And we didn't really have a strong entrepreneur in our team that wanted to say, yes, let's make a startup. We really were hoping that if we put out the protocols for making and using this method, and we also had an academic peer-reviewed publication. That's the other thing we forgot to talk about in our presentation. So both Sarah and we actually published small papers about these methods, and we contributed to a big review about the method too, with a whole bunch of academics. But that's not enough to make a product that somebody's going to want to buy and use or put out there. I mean, if people see, oh, well, it's already an open protocol, it's all available, then they think, how could I make a company out of it? And so that's how Fitz is like trying to break the mold. It is a hack to make up for the fact that we didn't make a Corona Detective company. At one time, GMO Detective had a .com web address, and we were thinking, okay, we'll just make a beautiful website, but then everything got slightly complicated because of the many different kinds of collaborations we had, especially when we were trying to get the clinical validation studies done. But yeah, what we've been doing right now is we thought that before we get regulators to officially declare this is a good test, we thought we should ensure that we could make batches that each time they were really reproducible and good, that the quality control was good. So we went the route of trying to make our freeze-dried reaction tubes via these big companies that are specialized in lyophilization. And we basically had one very exciting set of conversations with one company with that it went nowhere. And then finally, there's this Canadian company where we got our first proof of principle batch so far. And we have people in Ghana that are ready to do the independent validations that would okay the test for their FDA, the Ghanaian FDA. But then we still don't know yet if people in Ghana really want to take the test, to use this test. But then we got a whole bunch of other contacts. So we're doing a lot of communicating, like it's totally learning by doing. And there are people in different countries that are interested, but yeah, without a big pot of money, and with only, you know, these Zoom calls sometimes fall through. And so maybe Sarah, you want to say more too, from your perspective in America.
Yeah, I mean, like, exactly, Rachel, like you said, I know Ali Bektas has said the same thing, is that we thought if we make these diagnostic tests and they're sound, they're good, they'll be used everywhere. This was very naive, very naive. We have good tests that are sound and they're not being used, and, you know, not the way we would like them to be used. And it's very frustrating. It's extremely frustrating. And so we took a different route with the 1-hour COVID test, in that there was the company that approached us and wanted to license the test. And when I say license, we license it for free, because we wanted to get it to people for use. And we don't know about manufacturing a test kit, you know, and we don't know about marketing a test kit. And so this company says, okay, well, we can do all of that portion. And we had many discussions, especially with, you know, Tomas, about like, hey, we took some grant money, we received grant money to work on this as an open source project, but there's this company that maybe can get it actually in use. Like, what do we do with this? And we struggle internally a lot. And so in the end, we signed a contract with this company, but they, in the contract, we have a statement that says this test in its form that it is now can be used by any nonprofit or by any company that will do it at cost. And so this was kind of our compromise that wasn't a compromise on the open source idea, and could still potentially have this test get into use. So so Corona Detective and the 1-hour COVID test ended up taking very different approaches to how we thought we could try to get these still open source, but out to the masses. Right now, unfortunately, neither of us have been successful. I think
another thing about that is that, you know, at the time whenever all this work was happening, everyone just wanted to get products out to the market, and everyone wasn't thinking, am I competing against the giant incumbents that provide, like, government, what do they call it, the big the huge national testing? No, not Merck. No. It's one of the national testing. It'll come to me later, but, you know, the one, it's not LabCorp, one of the big ginormous US government lab, not US government labs, but US for-profit testing labs. They kind of ended up, I think, getting a huge amount of the market share because they had huge relationships with everyone. They were the incumbents, like they had the pipelines, they had the ability to scale up manufacturing. They mightn't have had as good a product as quickly as it was produced by the open communities, but they had everything else ready to go. We weren't thinking about that. You know, we were just how can we make something that'll improve outcomes for people?
It looks like Catherine wants to say something.
Yeah, I actually want to ease Sarah's pain a little on why it's a bad thing that we didn't get to the market as too quickly. I mean, I always try to see it from a more medical perspective, like when I'm a medic actually using this data or using this product, and what infrastructure implications does it have? Also, like, think about something simple as like digital incidences, you know, like when the testing lab needs to report to the government the data, and then it's a different test. And, you know, there are so many implications. And we are still in the middle of a pandemic, and like every government is struggling on so many levels. And to implement something fundamentally new in this phase is probably also not the right time, you know. So this is like amazing stuff which can help later on when we still have more knowledge about like this whole, yeah, huge building that we try to climb.
Yeah, we do talk about how this is work that's for the future as well. That is a bit of a consolation, but we need the pipeline, we need those capabilities, we need the ear, we need, you know, to gain the ear of regulators and players.
Can I say something? I think listening to both of you, can you hear me clearly?
We have some hard time listening to you. I think you will have some troubles with sound.
Oh, I got the head just because of that. Can you hear me at all?
No, the quality is really too poor, unfortunately. Could you write your question in the chat? No, you're muted now. So sorry for that.
I heard you earlier, by the way, in the previous talk. Something was working before.
Yeah, before it did work.
Yeah.
Can you hear me now?
Oh,
Yes.
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 Good. Okay, because I think this is quite important. I just give my feedback. I think you have to travel. I know it's a pandemic, but I think you have to go to Ghana, then see the Ministry of Health, and then make your presentation as you've done that. You have to take that chance and go to Ghana, make that presentation. Once you because the African Union is actually looking for solutions right now. So once you can have that, you can now take its steps, right? So you can now take that presentation probably to the African Union and see how you can now from there, then USID or the EU might be willing to fund the project. But I think that the first step is the first step. You have to go to Ghana and make your presentation, and then maybe in January or whenever.
Well, that sounds like a great suggestion, really. Our last call with someone from the Noguchi Institute, where they can do the independent validations, totally failed. We didn't get to talk to anyone, and maybe the only way to really make this happen is to go there. But we do hope that there is this, we have already at least talked to both the people from Noguchi who can do the independent validation, and to people from actually from Jenny Malloy's sort of network of people. People have done some of the clinical validations already with some of our tubes, but yeah, maybe we do need to just go there if our next call fails again. There's also a really interesting person who has something called the Health Keepers Network. And I was having this sort of fantasy that they were going to go from village to village with our corona detective tubes and like doing like in the village with the people doing the tests. But he said, no, no, no. They bring medical supplies around to different places, but there's no way that they're going to be doing the tests. So yeah, it's really a great.
Yeah, it's like you have the FDA, it has to be government approved. If the government doesn't approve it, even the health workers cannot use it. They cannot even test it, or else it would be a problem. And that's how fake news is spread. That there are some Caucasians that come in to take this and then another cycle. That's why there's a lot of caution, but it's something that I know I thoroughly enjoyed the presentation. I know that if you go to Ghana, make your presentation at the Ministry of Health, and then you get an okay, you can come to Nigeria, and then you just make a tour of Africa.
Do you know anyone at the Ministry of Health?
Like you've already started with Ghana, so once you have that go ahead, it's going to be easier for people like me to say, okay, there is a Moderna meeting happening this week. I would just send you in the Slack, so maybe you could attend. It's at the African Union, and then maybe you could attend that meeting, and then you could start meeting people, because you need to sell your product. Really, doesn't matter how good the product is, you still need to sell it.
That's the thing. I'm a biologist. I don't know about marketing, but no. We'll figure it out. That sounds great. Thank you.
This is something that JOGL Africa can help with. We have actually our local team based in Ghana. So this is something we could arrange. So we're almost at the end of the JOGL session. We have about 8 more minutes. Thank you so much for the discussions, by the way. It's and the feedbacks. It's been great. Would someone like to speak up again? And I've seen Vikram's comment on maybe inviting FDA regulators to the next Bio Summit. That would definitely be a great idea. We could potentially imagine also reaching out to them and create a special session, an open discussion session with some of those FDA regulators representatives, for example, to have their feedbacks on this project and record that so that we can share the outcomes of it with the community. That's definitely something that we need to organize. Yes.
Yeah, I think the FDA, this was such an unprecedented situation that the regulatory authorities were just so swamped. Remember they made decisions that, unless your thing is significantly better or beats these certain milestones, we're not approving it for EUAs, we're not even considering it for EUAs. And it was so liquid, everything was changing so quickly that they were just swamped. And like regulators, they want to help, like they're on your side, they want to give you the metrics and the milestones that we need to hit if the product is going to be allowed on the market. Like they're there to help. It's just hard to get to them sometimes because they're so busy. And if we had some kind of like a better relationship with a particular group in the regulator in each country, then maybe that would be a great idea. I know that in rare disease, I've been at some rare disease conferences, and and regulators have been quite happy to come along and give like they give presentations on what their state of the art is and what they consider to be the milestones you need to hit, but they're always very friendly anyway when you meet them in the real world. Yeah, Vikram, like, I mean, they always want early meetings. It's just a matter of bandwidth, getting access to them, because there's not as many of them as we need. And in my discussions with them, they're so smart, like they know everything because they see everything coming through. They just can't talk to you about it. So they're able to give you kind of directions and kind of very, very high level advice that is really beneficial to projects.
So probably we should start wrapping things up because time is running out, and we can keep the conversation going on in the Slack. And maybe more people will join us in JOGL. It's the project Nucleic Acid Amplification was our old project. Fitz is the new project to talk about, right?
So I just created a JOGL channel in the Biosummit Slack. So if you'd like to join it for continuing this conversation, you're welcome. And let me see if I can share a link directly from there. It seems I'm not super proficient to Slack.
It's getting late for you.
Yeah, definitely for Rachel too. All right. Yeah, it's just JOGL, exactly, because we've been discussing about so many things. So I guess we just fuse JOGL Fitz and the JOGL. Maybe we should actually differentiate the JOGL iGEM initiative from JOGL Fitz. Yeah, my brain is starting to fade off. So somebody will have to take over.
Yeah, thank you, everybody.
Thank you so much for your attention and your feedbacks. We'll try to organize an open session very soon again, and we'll just let you know. And if you're interested in following up, please do join the JOGL platform. And there is a JOGL Fitz space that you can join. And we'll take it from there.
So today we're having this lightning talk that unfortunately for our speakers are not going to be presented. So they're going to be presented in a pre-recorded fashion. So from now, without taking any more time, I would like to present the first speaker, Oluwadamiola, Ogunjobi. And if you're already around, please step up and the microphones are yours. Let me stop. Oh, sorry. Let me stop my sharing my screen from here. And Ola, if you're around.
Hi, everyone. So I would be presenting on the impact of biohealth relations in biopublic relations and biohealth security. Building trust, health security, health care as a commodity and marketing information. So public health relations simply means, can you hear me clearly? Because I am trying to make sure that I'm heard. So public health relations simply means marketing information. At the inception of the pandemic, or pre the pandemic, no one even considered that public health security would need public relations. Although there's been a lot of research, and when you go through my presentation, you find a lot of research that has been done, but not to this extent that everything could be considered fake news. So history and civilization has it, that's the Greek, the Roman empires, and the world wars, has it that diseases were weaponized. And because of diseases, that's why World War I, World War II came to an abrupt halt. Okay, let's say World War II was because of Nazi Germany being invaded. But the consideration of World War I is that because of the amount of people that died of various diseases, which include malaria, typhoid, smallpox, that's why the war had to end. So from inception or time memorial, wars have been, diseases have been weaponized. And so studying why public relations is important, it is imperative at this time to understand that for purpose of, I'm a bit tired because it's about almost 12 midnight here, so please pardon me. For the purpose of biosecurity, there has to be strategy assigned for government, for institutions. Government, when I say government, I'm talking local government, state government, and the federal government, understanding that all states, even though we all live in the same state, we do not have the same values. So the way they have to guard this and measure it is to look at it through the lenses of geopurpose, because not all states have the same religion or values or even diets, the kind of food that they eat. So because of these reasons, it's imperative for states to start to look at the purpose and the strategy. Then the states have to also look at the infrastructural designs. In a country like from Nigeria, you can actually take a trip from Nigeria through Niger to Mali and get to Liberia. Libya rather. And you have to consider people mobility in this purpose and strategy design. The fact that when the COVID-19 broke, the porous borders along these regions, these regional boundaries, and people were infected and reinfecting people, the messaging to these people cannot be a one size fits all. If the moment you start to tell people, okay, wear your mask, they say, why? Why do I need to wear my mask? Okay, the people in Nigeria, they're not wearing their mask, or people in Lagos are not wearing masks, and the death toll is not as high as people in Zamfara state. So if the government or the policymakers are not able to have in-depth examinations of the infrastructure that is available, that's the institutional infrastructure, we're looking at the healthcare, that's the health system available, we're looking at if the PPE is available, we're looking at if the people that the health workers available, if they even have the amount of training that would help contain the pandemic. So that's something that is important. Then the next would be comprehensive risk reporting and effective communication. At the beginning of the pandemic, the WHO did not give, I wouldn't say they didn't give the right information. They just gave information that this disease is not contractable. And because of that reason, the Trump administration, with its political agenda of not wanting to lose the 2020 election, went on a crusade and started to bombard the airways with different information. And so because of that, there was a lot of panic and there was a lot of confusion. And because of this problem, people from Asian communities were attacked. So public relations in biohealth security is very important. In fact, it can decide whether the pandemic will be contained or whether it would just spiral out of control. The next will be disease surveillance. Are we using traditional disease surveillance as tools? That's the measuring of biopreparedness in the area and trying to forecast. Are we using non-traditional with the AIs, etc. for this? But at the same time, if this instrument, and I think I mentioned it to the other presenters, if these instruments are not properly, they are not properly introduced to the community, you would hear, oh, they want to draw your blood, they want to draw your blood for this reason, this, and so people become biased, and then there'll be misinformation, there'll be fake news, there'll be half news. As regard the origin of the pandemic, recently, some researchers said maybe the pandemic originated from a wet market, and they went ahead and did the research. But the average mind would believe that this pandemic was originated from a lab. And because the inception, the first information that was received from I'm not blaming the WHO, but that first information and then the political backing that was not addressed immediately is really what, in my opinion, in my honest opinion, is what really exacerbated this pandemic. Because we had the Ebola pandemic, we had the Ebola crisis rather, we had Ebola in West Africa, but even right now, the information.
I'm sorry, Damiola. I really would like to continue listening to part of the talk. Unfortunately, we have only 10 minutes for every speaker. And, yeah, and well, I really appreciate. I think so. I think it was already like 2 minutes left, maybe.
Okay, let me quickly wrap up.
Yeah.
Okay.
So
technical assessment of information. So recently, Big Bird was in the news. Why was Big Bird in the news? Because someone decided to roll out vaccine. What's that word? That there's a word for it, and I'm trying to hurry up. That vaccine to enable people to get vaccines. That was why they decided to roll out this public relations ideology. But it backfired rather, because they did not consider that there are other groups that will see this as propaganda. So where does public relations start or end, and propaganda start? So for this reason, it's imperative to start to, let me go to the last slide. It's imperative to start to carry the entire stakeholders, carry them along. Recently, there's a mandatory vaccination for the military and the police. And this it's not a bad thing, but if these institutions that protect us are not carried along, you can imagine what will be going through the minds of regular folks if the military is literally, let me not say forced to take the vaccine. And I wanted to say something earlier. I said, so there has to be a difference between vaccine hesitancy and then the active and anti-vaxxers. This information has to be properly explained on a continuum.
I'm sorry, but we must.
Yeah.
I'm sorry, but we must proceed. But thank you so much for your talk. And everyone, if you're interested, please continue discussion in Slack. Thank you so much.
Thank you.
Okay, thank you, Xavier. Thank you, Oluwadamiola. Yeah, unfortunately, we're running just for 10 minutes every speaker. Our next speaker is Gary Lansman, presenting ECLIA, a methodology for determining COVID-19 antibodies. And Gary, if you're around, please step up, and the microphone is yours.
Okay, thank you. Let me see if I can share my screen. Am I able to share my screen for the slides?
Oh, yes, you should be able to now.
Can you see my slides or no?
No, not yet. Click the for within Zoom. Click the green button for share, and then you'll be able to. Yep. Then you'll be able to find your PowerPoint, and then click the window that has your PowerPoint, and it should bring you up on our screen.
Thank you.
You're welcome.
Now can you see it?
Yeah.
Okay.
Yes, indeed.
Okay, perfect advice. Well, welcome everyone. My name is Gary Lansman, and I'm a member of the Counterculture Lab Biocurious COVID group in the San Francisco Bay Area in the United States. And this community discusses everything COVID related. My goal in the next few minutes is to describe one test used to detect antibodies to the SARS-CoV-2 virus that causes COVID-19. And there are two reasons you're going to have these antibodies. The first is you've been vaccinated, and hopefully this is the case. Your immune system has mounted a response to the vaccine producing antibodies. The second is you've been infected. And unfortunately, you are among the approximately 277 million people worldwide who have been infected. In this case, your immune system has mounted a response to the virus itself. The test I will describe is an electrochemiluminescence immunoassay, ECLIA, ECLIA for short. In particular, I'll be describing the Roche diagnostics test. It received FDA emergency use authorization on May 2nd of last year. LabCorp and other laboratories in the United States offer this test. It's also used in antibody research. It was used in an Israeli study of breakthrough infections. It's currently being used in a study of immunocompromised cancer patients in the United States. As you may know, the quality of a test is measured by its specificity and its sensitivity. For this Roche ECLIA test, the specificity indicates how well the test identifies those without antibodies to SARS-CoV-2. Roche publishes that its specificity is 99.98% for blood samples taken 14 days or later after vaccination or infection. Sensitivity measures how well this test identifies those with antibodies to SARS-CoV-2. And Roche publicizes that its sensitivity is 98.8%. From your vaccination, or in the unfortunate circumstance of infection, your immune system responds. Your adaptive immune system's B cells are activated, and they produce the antibodies that this LabCorp antibody test measures. LabCorp calls its test a SARS-CoV-2 semi-quantitative total antibody spike test. I'd like to unpack this name. What does semi-quantitative mean? Simply, it's a numeric scale for the results from this test. A qualitative test only provides a positive or negative result. What does total antibody spike mean? It's all the antibodies that your immune system produces, and there are three: IgG, IgM, and IgA. How does this LabCorp ECLIA test work? First, a phlebotomist will take a blood sample from you and send it off to the lab. In the lab, there is a first incubation of your blood sample. It is mixed with a recombinant protein mixture, actually 2 mixtures. These lab-developed recombinant mixtures match viral characteristics of the SARS-CoV-2 virus, particularly its nucleocapsid. The spike and nucleocapsid protein of SARS-CoV-2 are commonly used target antigens in COVID-19 blood tests. The spike protein projects on the outside of the virus membrane, and the nucleocapsid encapsulates the viral RNA within the virus. Both are immunogenic. They provoke an immune response. The purpose of these lab-developed mixtures is to bind with any antibodies in your blood sample. Of the 2 lab-developed mixtures, one mixture is labeled with biotin. Biotin will act like an immunological anchor after the mixture it's in attaches to your sample's antibodies, and I'll describe this in a little more detail in a minute. The second lab-developed mixture is labeled with ruthenium. When the ruthenium mixture attaches to your sample's antibodies, it will play the role of a biological flashlight when perturbed. Let's look at this in a little more detail. Okay, we've started preparing your blood sample so its antibodies can be measured. We've added 2 lab-developed mixtures to it. They bound to your antibodies, if any, and now a second incubation is required. Streptavidin microparticles are added to your sample. They bind to the mixture with the biotin, now in the combined mixture. This combined mixture is placed in a measuring cell. In an analyzer, a magnetized electrode attracts the streptavidin anchoring your antibodies. Unbound material is then washed. The analyzer discharges an electric charge into the measuring cell. This causes chemiluminescence in the ruthenium bound to your antibodies. The analyzer's photomultiplier captures this chemiluminescent emission. This provides a measurement of your antibody titer. The higher your antibody titer, the more chemiluminescence and the higher your measurement. The unit of measurement is in units per milliliter. Roche has established a threshold for COVID-19. If your measurement is less than 0.8 units per milliliter, the result is negative, indicating that no antibodies were detected. If your measurement is greater than 0.8 units per milliliter, then the result is positive, meaning antibodies were detected. Healthy individuals typically score above 100 units per milliliter. Although not a gold standard for COVID-19 antibody testing, this ECLIA test does provide one measurement of the level of neutralizing antibodies that you have from your vaccination or infection. Having a numeric scale, it's better than a qualitative test, which would only, pardon me, which would only provide a positive or negative result. As I conclude, I'd like to mention what this PET test does not do. The result is not an exact measure or number of antibodies. The test will not tell you if you have active COVID-19. And as shown here, the FDA states that results from currently authorized SARS-CoV-2 antibody tests should not be used to evaluate a person's level of immunity or protection from COVID-19. This test does not measure other immune system actors like memory B cells and T cells, memory T cells. The research into levels of immunity and protection provided by antibodies is ongoing, and the FDA has not issued any guidance for interpreting results from this test or others. So, David, this completes my description of LabCorp's ECLIA test for antibodies. I think Mohammed may be next.
Yeah, thank you very much, Gary. Well, we have 30 seconds for any question from the audience if they want to have like a very, very quick question. Nobody?
Oh,
or maybe we can wait for the wrap-up session.
I had a really quick question.
Okay.
Is there a more detailed paper or PDF of the ECLIA, or is it strictly a slideshow or a concept that you've talked about on the Zoom calls and the COVID-19 chat rooms?
I will mention that on the slides, David, which I assume are posted, there is a list of references. So if you can get access to my slides, at the end, you'll see a list of references.
Oh,
but
okay,
there's no PDF of what I just mentioned. No.
Are the slides on the Slack, or are you going to post them in the chat?
I guess we can upload. Oh yeah, fantastic. I guess we can upload that into the Slack. And yeah, I would like to pass to the next speaker, please. Thank you very much for.
Thank you, Gary.
I'm really sorry for the Thank you, Gary. I'm very sorry for the short time. Our next speaker is Mohammed Atif from STEMcell, Pakistan. And
hello, everyone.
Mohammed,
am I audible?
Microphones are yours.
Am I audible?
Yes.
Okay. So can I share my screen?
Yes.
Okay. All right. Let me share. I think my screen is visible now.
Yes, it's now visible.
Okay, so let me. My screen visible or not?
Yeah, the screen is visible.
Okay, so hello everyone. Good morning, good afternoon, or good night. I don't know, it's almost 3 a.m. right now in Pakistan, and I feel a bit sleepy, but here we go. So before I get started with my presentation, so this is me, Mohammed Atif. I'm a co-founder and director of STEMcell Pakistan, and I introduced to this genre to work for the popularization of the science. Back in 2016, I started a YouTube channel. I make videos on different science topics, and then as a side hustle I started, but I really get interested in it because I had a great chance to interact with the like-minded people, and it's my second time at Global Community Bio Summit. So it captivated me because this global community always motivated me, inspired me. I heard a lot of the stories, and I get this idea, the taking actions, it's important because for the sustainability of the society, your small step can make an impact. So, let me give you a real quick story of the STEM education, what's going on in Pakistan. So there are two types of the system operating when it comes to education. There are private organizations, institutions, and schools and colleges and universities, and the next are the public school, colleges, and universities. So like, you know, the students, they mostly come from the middle class income families. So they get enrolled themselves in the public institutions, but they are not well equipped with, you know, a right quality of when it comes to curriculum or when it comes to facilities. And also in the next figure, you can see, like, you know, the figure goes up and then it starts declining. And amid the COVID-19 pandemic, when the educational institute closed, so there come a vacuum created in the educational system, especially you know, for the students hailing from the underprivileged areas. So this create a huge vacuum. So amid the COVID-19 pandemic, I had a privilege to be a part of fellowship, AML fellowship. Let me give you a brief overview, a brief introduction to the AML fellowship. AML fellowship is an Stanford University sponsored and funded fellowship for the Pakistani students. So being a part of the AML fellowship, I submitted my proposal for this citizen STEM portal, to launch a citizen STEM portal for Pakistani students. And so our main vision was to create a portal and, you know, and open source learning. With a mission, like STEMcell Pakistan is an organization, it's a non-profit body aimed at development of an open source society as a citizen STEM portal to promote STEM education in the idea of skills future among the masses by providing platform for cross-disciplinary research and collaboration to bridge the gap between science, society, and technology. And we are a team of a diverse group hailing from multidisciplinary fields of science and technology, and we are optimistic and we are on a journey to capable Pakistani students, especially the high school students, to become a lifelong learner in the field of science, technology, engineering. Our main objectives and are actually to educate the young girls. You know, basically in Pakistan, girls are more inclined towards the art education, and it's just because of the lack of the awareness towards STEM education. And so we are our main one of our aim is to educate the young girls with, you know, about the STEM education, and to work for the popularization of the science and technology in low income schools that lack the quality of the education. Also to aware the public sector, those students who are from the public sector institutes or from the underprivileged areas about the opportunities in pursuing STEM education. So already we are making an impact. We are in touch with national level NGOs and institutions, and also 20 plus members are in our team from different part of the country, and 300 plus volunteers are signed up for our cause, and they are a part of this growing community. And recently we had done a STEM boot camp. It was a virtual activity, but the response was overwhelming. We had a participation from different sectors, from different part of the country. We aware the participants about the career prospect in STEM and other related stuff related to STEM. Just want to thank the few main members and the stakeholders of this STEM portal. STEMcell Pakistan. Salman Promon, he's from Bangladesh. He's a part of the Global Community Bio Fellowship as well. He helped me a lot in, giving me the overview, like what I have to do for the for overall for this STEMcell Pakistan. And Sameer Ahmed is a co-founder. He's running a organization, Young Scientists Union. We're working on infectious disease like diabetes. And the third one is Shugaftar Ahmed. She's a research assistant at Precision Medicine Lab and also co-founder of Gapi, which is actually a health sector startup. Also, we built actually a model to, way forward and to push this startup further. So we are thinking to first assess the situation in different parts of our country, like what is going on actually at different part, and then we have to develop the strategy around it. And based on that strategy, we have to implement and we have to adopt. And then we have to look around for the collaborations to reach out to institutions or NGOs and non-profit bodies who are working on the same genre or who are interested, to promote this STEM education. And then the last step is to spread the awareness. Announcement part, like this is a very special thing for me, because we and our team decided to launch a STEM regional fellowship for Pakistani high school students, and we are looking for support from the global community. Definitely, if anyone interested in for support, any kind of support, we are looking forward to it. And you can reach out to us on Facebook, Twitter, Instagram, or LinkedIn. And also, I'm going to put all the social media handles after my presentation in the checkbox. Feel free to reach out to us for any type of support or any kind of feedback you may have. This is our talented team.
Thank you very much for the presentation. I'm really sorry that we have to be very quick. Please thank Mohammed, and if we can follow them later on Slack. And unfortunately, well, I will have to present our next speaker that is going to be in a pre-recorded fashion. So our next speaker is Kahlil Corazo from Accessible Genomics in the Philippines, and he has prepared a video for us. And I'm going to share my screen for presenting his work.
Thank you, Mohammed.
Thank you.
How do you do the dirty work so that the scientists can do more science? In this talk, I share 3 playbooks to answer this question. My name is Kahlil Corazo, and I am the project manager.
David, I cannot see your screen.
Genomics. 2020 was a terrible year, but it was also very inspiring. We knew that we needed genomic SARS-CoV-2 surveillance, but we did not have enough sequencers in the developing world. Project Accessible Genomics was a call for help, and we received so much help from scientists and students from around the world, from companies and the open science community. In 10 months, our partner lab sequenced SARS-CoV-2 in the region. My role in that project was to do the dirty work so that the scientists could do the science. Over the years, I have collected playbooks for this work. I share my top 3 in this presentation. First, for known problems with known solutions, project management. Second, if I'm starting a business where the problem and the solution are not completely known, I use evidence-based entrepreneurship. And third, if both the problem and the solution are currently unknown, I increase my luck surface area by using the playbook I call designing serendipity. Okay, first, project management. We knew both the problem and the solution with project accessible genomics. We just needed to bring a low-cost sequencer to a lab in the developing world and organize trainings for them. Instead of one giant monster of work, you break down the project into manageable components using tools like the work breakdown structure to manage scope, or the risk register to prioritize risks and plan responses. To learn more about professional project management, just go to pmi.org. The Project Management Institute is the biggest association of project managers in the world. And I recently wrote a book entitled Professional Project Management with Rome, which you can read with the app Rome Research. Second, evidence-based entrepreneurship. Through Project Accessible Genomics, we uncovered a more fundamental problem in doing molecular biology in the developing world. The biggest threat to the project turned out to be access to reagents. They are expensive for us because we need to buy them from the US or Europe. Then we need to pay for a cold chain. Then we need to pay local gatekeepers, both legal rent seekers and sometimes corrupt ones. What if we can manufacture some of these reagents locally? Then we can bypass all this obstacles to science. Now is the time for this because of advancements in open source wetware and low-cost hardware. We need a self-sustaining solution where we have sufficient customers to pay for the manufacturing operation. We need to prototype both the manufacturing and the sales and marketing machine. For the latter, we are employing the Lean Launchpad, or evidence-based entrepreneurship. This playbook was created by Steve Blank, a Silicon Valley serial entrepreneur and now teaching at Stanford. This playbook is now used in the National Institutes of Health, or NIH, of the United States, and accelerators like IndieBio. The best introduction for this crowd would be Steve Blank's interview with Arvind Gupta. You can also sign up to my Substack, Bioentrepreneurship with Garage Door Open, where we DIY the Lean Launchpad to build a local manufacturing operation of basic lab reagents. So lastly, designing serendipity. Instead of being the worst year ever, 2020 became for me one of the luckiest, and it was by design. In the past years, I've collected techniques to build a playbook, which I call designing serendipity. One of the key ways to expand your luck surface area is to work with the garage door open. This is why I'm sharing with you what we are working on and these 3 playbooks. To know more, visit designingserendipity.com. Someone needs to do the dirty work so that scientists can do more science. If you're the person, let's help each other. Please reach out.
Well, that was Corazo from the Philippines, and our next speaker is also presenting his talk in pre-recorded format. And let me share the screen again. And this is Tanishka Kumar from India and the Modi University.
I'm Tanishka Kumar, studying BTech Bioengineering. I'm currently doing an internship on.
I need everyone to mute their mics. There's an alarm that's going off. Thank you.
I'm also a part of the Rhizomatic Review Journal as Amber. So when I was quarantined for around a month after being detected for COVID, I was curious, as many of us, about the steps our countries were taking to combat
Yes, David, I cannot hear the audio. Can we replay from the beginning?
Information technology facets with my group. Steps our countries were taking to combat the COVID outbreak. Not being able to attend my classes, I was searching for a community and chanced upon Biosummit. So here I am, speaking on India's response to COVID-19 phase 2 through government initiatives, research, development, bioproduct development, information technology facets with my group.
Hi, I'm Ajli Singh, pursuing BSc Honours Biomedical Science third semester at the University of Penn. As of now, from the subjects that I have been studying, I have developed interest in biochemistry.
Hi everyone, I'm Rimji Marora, studying BTech Biomedical and have an interest in development of instruments for better treatment of various heart diseases. As conveyed by Tanishka, we will discuss each facet one by one. Please continue to watch if you are curious about what the world's second most populated country did to grapple with COVID-19. Given here is a graph which shows record of deaths due to COVID. On April 28 this year, India recorded more than 3,000 deaths in a day and reached the 4,000 mark on May 8.
So let's start with the government initiatives. Contact tracing was one of the methods used to prevent the further transmission of the virus. Like any other country, RT-PCR tests were made mandatory at public place entries like airports. The identified COVID positives or the people who came in contact with them were directed by the officials to quarantine. Now, the COVID-19 communication strategy served as a key guidance to the state program managers and partners to plan and implement the strategic approach to address incredulity for the vaccine, sustain the demand, and support the maintenance of the COVID-appropriate behavior. It includes strengthening mass media, social media, engaging international media, social mobilization, and community engagement. Link has been added for further information on the same. India launched a multi-pronged nationwide mass awareness program to burst the myths about COVID-19 vaccination program organized by Indian National Young Academy of Sciences, carried out through Kovac News, which is an Android-based mobile app, and a pan-India infographic video and audio competition with the motivation to transform information into better readable, effective formats was also organized along with the Gyan Tika webinar series, dedicated to increase the awareness about the vaccines through eminent speakers and open statement on COVID-19 vaccination. CoWIN is an Indian government web portal for COVID-19 vaccination registration, owned and operated by India's Ministry of Health and Family Welfare. It displays booking slots of COVID-19 vaccine available in the nearby areas and can be booked on the website.
COVID response framework was released by the Department of Biotechnology under research INSACOG. Indian SARS-CoV-2 Genomics Consortium is a multi-agency lab set up by the government. It was for the sequencing and analyzing the genome data to understand how SARS-CoV-2 virus spreads and evolves. Testing included developing molecular and serological tests, development of novel diagnostics, testing hubs, mobile iLab. Treatment included plasma therapy, monoclonal antibodies, drug repurposing, and DBT Ayush phytopharmaceuticals. Genomics included INSACOG, and there was nasal microbiome study, and INSACOG was successfully completed. Prevention included multiple vaccine platforms, assay animal models, clinical trial sites, and immunoassay labs. To facilitate the lab and facilitate everything, included 5 COVID-19 biorepositories, indigenization of resources, validation of centers, and rapid regulatory framework as the
National Institute of Immunology, point of the serology test, commentary at GIGM and GCBS. Vaccines like Covishield, which was prepared as Serum Institute of India in AstraZeneca vaccine developed at the University of Oxford. Now, let's discuss about therapeutics. Various therapeutics were done. First, use of blood plasma from already recovered COVID patients using the lymphocytes obtaining desired neutralizing antibodies against 2019 and COVID. Second one, DVT Birex supported antiviral drug Biorafine by Zydus Cadila has been approved for emergency use of moderate COVID-19 infection. Third one, DVT Network Project with Ministry of Ayush for screening of 15 medicinal plants and 8 additional Ayurvedic formulations for anti-SARS-CoV activity. Fourth is DVT ICGED, successfully generated panels of human monoclonal antibodies against SARS-CoV. Various startups also developed therapeutics, and these startups were supported by Biolabs. Websites were created that made information access easier. Self-taught developers, experts from IT sector, state government, and others joined hands to create an application and website to help people sort out real-time vacant beds, oxygen cylinders, vaccine details, and mental health counseling.
Rashtrayak, Jaipur-based software startup, launched Rashtrayak Toolkit apps that sources out needful used information, blood banks, oxygen ventilators, and vaccines availability for those living in rural areas.
There's 3 things you can take from this presentation. Government initiatives like vaccine campaign were huge success, while contact tracing wasn't that effective or wasn't properly implemented. Research and development of biotech products, websites, and COVID guidelines developed by institutes and organizations reaped good results. Mental health of the citizens could have been prioritized more. Now, India and its citizens is still going through a lot, and there's struggle, but there's improvement that cannot be dismissed. The following slide will show our contact information. We can contact each other, get in touch, and I would love to further the discussion for these topics and more. Thank you.
Okay, that was Tanishka Kumar and Ramesh Arora and Anjali Sain from India, the Modi University. So the next speaker is Castillo that he also sent the pre-recorded format, and here is the video.
Hello, I'm Kazutoshi Suda, researcher in Yamaguchi Center for Arts and Media, commonly known as YCAM, that is an art center located in Yamaguchi City in Japan. Since its opening in 2003, YCAM has been hosting a variety of events including exhibitions, performances, movie screenings, and workshops for children, operating around a central axis defined by the pursuit of new artistic expression incorporating media technology. In 2015, we took part in an online course titled Bioacademy, also known as How to Grow Almost Anything, and visited domestic and overseas academic institutions and laboratories to establish new biolab and start our bio research. We began exploring a variety of points of view aimed at seeking the application possibilities of biotechnology. We have opened up our activities in a variety of forms: research showcase, to introduce the process of research to the public; workshop, for wider generations to understand living things and ways of examining them using hands-on experiments; intensive workshop, to explore the application possibilities of biotechnology in terms of reading, writing, and bioethics; exhibition, in collaboration with artists, and the release of publications such as books and magazines. From our various activities, in this presentation, we will introduce about YCAM Open Lab 2020, Understanding Living Culture 2. YCAM Open Lab 2020 is a series of online talk sessions that reflect on the state of community biotechnology, citizen science, and arts for responding to COVID-19. In previous Open Lab, in 2019, we welcomed members of organizations with active biolabs based in various regions in Europe, Africa, Asia, and Oceania. In 2020, we moved our venue online, welcomed our guests one by one each week, and hold a talk session discussing the situation of where their activities are based, as well as their ongoing initiatives. By interacting with biotechnology and art practitioners, we shedded light on the role of biolabs, their relationships with life and culture, and further possibilities of public facilities. As the title mentioned, this session took place in 2020. Although it was recorded around May to August 2020, when the world was working on the early response to COVID-19, the content is still relevant and thought-provoking. First session is introduction. The theme is response from open source and DIY community to COVID-19 from us. Second is collaboration, community, and jamu. 3 elements of COVID-19 prevention in Indonesia. Talk with scientific researcher Nur Akbar Arofatullah. Third is COVID-19 prevention in Ghana. Open science and biotechnology. Talk with Harry Akligo from Hive Biolab. Fourth is biology, humility, and possibility of art. Talk with Oren Katz from Symbiotic A in Australia. Fifth is citizen science, community biotechnology, and augmented ecosystem. Talk with wild scientist Kosuke Katano. Sixth is Biolab, artistic platform and access for public. Talk with artist Georg Tremmel from Bio Club in Japan. Seventh is being artist, scientist in COVID-19 era. Talk with biohacker and artist JJ Hastings from US. Eighth is pandemic and citizen initiative. Trust, transparency, and open access, with Bess Lee from GovZero in Taiwan. And ninth is arts after pandemic, solidarity and constituencies. Talk with curator Bojana Pisker from Slovenia. A wide range of talks have been recorded, from reactive responses that require urgency and expertise to preventive proactive stances in daily life, and from science and technology to art and civic initiatives. All 9 sessions are now available online, and we hope you will check them out. Thank you for your attention.
And that was Kazutoshi Tsuda from the Yamaguchi Center for Arts and Media. And our last speaker in the pre-recorded sessions is going to be Lucas Potter presenting Consumers of the Pilot Study, and his video is here. Oh, excuse me.
Hello everyone, it's nice to meet you. I'm Lucas Potter. This is the Consumer Surplus Pilot Study. I'm presenting this work on behalf of myself, Dr. Oluwadamiola, and Xavier Lewis-Palmer, all from Old Dominion University. So, as we all know, that during the recent COVID-19 pandemic, a lot of communities went into lockdown or quarantine. Because of that, some of their behavior shifted, particularly online behaviors. But we found that the metrics of overall service use or metrics of overall service quality, like a basic survey that just said if you liked an online service or didn't like it during lockdown, were flawed. They weren't really actionable. If you gave them to a provincial or local government, they couldn't do much with it. So we decided an actionable measurement, behavioral change, and internet-based behaviors is needed for quantifying the changes of online behaviors during quarantine. So here's what we did. We found the consumer surplus in internet-based values before and after quarantine. We did this by asking the following questions you see here in quotes. And this question results in 2 values per service for COVID before lockdown and after quarantine or after lockdown. The survey was distributed or disseminated to the only population that readily available, which was college visiting students ages 18 years and older. The survey was given by Qualtrics XM. That's not an endorsement of the service, but just what we had. And this was experiment was rated as not requiring full IRB review because we didn't do anything invasive with participants and we never collected any personally identified information. So to analyze this data, what we did was found the consumer surplus before quarantine, directed from consumer surplus after quarantine. This helped us. This helped us find the exact values of how much money people were willing to spend on a service when they were locked down versus when they weren't. Found there's average results for every service, found the standard deviation, and we compared the results by gender, and you'll see why we did that later. Results. So here in the blue bars, for every service you see on the x-axis, you see the pre-lockdown or pandemic ratings for consumer surplus. So for instance, people would say that, oh, I'd be willing to stop using a cell phone for 30 days only if you gave me around $550 before the pandemic, and that rose to around $650 after the pandemic. And of course, we have different services here like cell phones, emails, all the way to Amazon or Netflix and YouTube. So what we did from this is first we found differences between these two values. So you can see the x-axis is exactly the same. This y-axis is now the monetary monthly consumer surplus for each service. So you can see the three big standout values we had that required more than or represented more than a $200 difference was video chat, Netflix, and YouTube. This third result was just very interesting because YouTube is a free service, so presumably people aren't willing to pay for it because, well, they don't pay for it, or rather they are ad supported. But yeah, people would need, on average, our population would need a very large amount of money in order to part with YouTube during the pandemic. And of course, you have all the other services here, and we'll get them later. The other big thing we did was split this up by gender, identify differences. You can see the error bars are much bigger this time because we're using many fewer people in each group. You can see the only real thing that we have a huge difference in is video call or email here at the very beginning, food delivery, and Netflix. No idea what that could be. But let's get to the weaknesses because there are many in the study. It's only a pilot study. The population was composed entirely of engineering degree students from a single U.S. university that's good access to. They were 75% male identifying, so we can definitely have a gender bias in this study, and less than 70 total respondents. It's not a very large study. Future directions, we would like to do a wider survey with enough participants to have results separated by socioeconomic strata, and this will help identify needs of those most impacted by quarantine measures. As we saw, people who tended to have less resources, in the US especially, tended to not do as well during the pandemic, either in direct measures, as in their quality of health, or in indirect measures, as in how many hours they had to work just to keep by. Thank you. This work was brought to you through Oldham University in Norfolk, Virginia, and Biolabs, which is also located in Norfolk, Virginia. I'm very happy you guys came by to see this presentation, and if you have any questions, please contact us at lpott005@odu.edu. Thank you.
Okay, and that will be our last speaker presenting his video. And I believe that we have like 8 minutes left for any of the questions that the participants already here. If anyone wants to follow up, or if yeah, I mean, we can more or less save this last 5 minutes or 8 minutes.
Home open PCR.
Oh, sorry, then you have one question?
You want to hear about qPCR testing at home?
Oh, yeah, I guess that will be Gary's. Yeah.
Oh, okay, I just wasn't sure when.
I'm sorry, could you repeat your question?
I was just wondering if you have time for the open PCR testing at home for COVID.
Yeah, some of the audio cut off. I am sorry. Could you repeat one more time?
About PCR or open PCR testing at home for COVID.
Yeah, the first part cut off.
Oh,
could you type it in text?
Yeah, we're really sorry. I think the audio has been playing a little bit of a bad joke for us too. Yeah, I think you were referring to one of the talks earlier. It's my understanding. It is within this track.
Oh, okay. It's okay. I was just wondering if you had some time to fill. We could fill it with that.
Oh, okay. Yeah. Well, definitely. The thing is, we have like 6 minutes left from this point.
Oh, okay.
And
That's fine.
yeah, definitely. After that, we can go a little bit further in the discussion for the panel if you want to elaborate a little bit.
No, that's fine.
Yeah.
That's fine. Thank you.
No, it's okay. Is anyone else that want to direct to our previous presenters, our previous speakers, Mohammed or Gary, the 2% here, they want to address them any further questions? We have like 5 minutes left, and I guess it will be like great opportunity to know a little bit more of what they're doing. No one? No? Come on, don't be that shy. Okay, if that's the case, I guess we should move on. And we have a panel that it was suggested by Don. It's an open discussion about open COVID-19 detection protocols. And I don't know, Dan, if you would like to go further a little bit in trying to give us a little bit of context on what are your ideas when you propose to the panel. I know that you suggested to review some of the protocols that are currently used for COVID-19 detection. And I don't know if you can give us a little bit of context.
Okay. Would you let me share my screen? I have a quick PowerPoint that might help explain it.
Sure.
Okay, thank you.
Share away.
Can you see it?
I know it's not coming up yet. Yeah, not yet. Click the green icon. Find your PowerPoint of interest. Click that window and make sure that you have share sound selected. And then click blue share button in the lower right corner.
It's freezing up on me. Okay, well, I think it's freezing on me, so sorry. Okay, so the different protocols I used, I've got a qPCR machine that I got in the beginning of the pandemic. It's called a Mini 8. It's from China, from Coyote Bio, and I got it off eBay used. So I first initially wanted to use just PCR, but then I realized that you couldn't really quantify anything with PCR. You could just detect whether it was there or not, or how much, very easily. So we just decided to use qPCR, got a machine, and then we kind of had to teach myself how to use it. And so that wasn't easy. So there's a few steps you need to extract. So you have a little swab for the nose, but then I realized that it's easier to test saliva. And according to Professor Iwasaki from Yale, who's done a lot of research in this, there's a lot of protocols that can use saliva just as effectively, maybe more so. So I decided to go that route rather than use the swabs up the nose. But in my case, we considered safety as well. I mean, I was capable of testing for it in the end, but I didn't really have any symptoms of my own, and I didn't want to bring in COVID into my house because our lab was closed, and probably shouldn't bring it to the lab anyway, but there weren't enough safety precautions to bring a virus that was live or even in a vial into my home. So I decided just to test myself and run the protocol on the machine. So I ordered the primers from IDT, which were approved for research use only. So I got the cheaper primers for about $100. So I got about 50 tests out of those $100, so fairly cheap test to run. And then I ordered from Thermo Fisher. I don't know if any of you belong to it, but there's an Aspire program, ASPIRE. And in that Aspire program, you get one free product a year. So I got a free product, and that was a probe for COVID-19 that was approved by CDC. So I tried that one and came up with mediocre results. The curve wasn't very pretty on the qPCR machine, the results. So then I tried QuantaBio, who sent me some free samples. And the beauty of QuantaBio is you don't have to, in the extraction process, you want to process it. And some people use Trizol and other preservatives and things to stabilize the RNA. But if you look at the CDC's protocol, you don't have to do that. With QuantaBio, if you're in a resource poor area that you don't have access to all the enzymes.
It's quite long, so check it out on Vimeo. It's called Water Warriors. And
okay.
Is anyone please turn off?
Okay, that's
mic.
fine. So the QuantaBio, I ran that, and it actually worked very, very well on the positive controls sent by IDT, and the negative controls were flat. So it came up with a very nice curve. So basically I determined QuantaBio was the way to go, you know, and a portable PCR machine such as this is, get closer, is the way to go too, because it's portable. It can run off batteries and can be used in countries or remote areas where there isn't a lot of scientific equipment. And it's quick, it only has 8 wells. I've since purchased at a bargain rate in an auction, open qPCR. So you have 16 wells as opposed to 8 in this one, and that would be better. I'm looking forward to using that one as well. So that's my experience testing at home. I think it was a success, but again, I didn't have the virus that I'm aware of, so I didn't actually get to verify it 100% and wasn't willing to bring in a live virus into my home.
Dan, in your experience, what could be some of the limiting factors for setting up a test like that at home or any other remote facilities?
Okay, well, you just need a few things. You need the IDT to send you the reagents, the primers and probes, and the negative control. I ordered a plasmid, which was a positive control, which has the whole N capsid gene in the plasmid, which is great. So then you just test against the N plasmid. So, you know, it's not really money because it's only, like I said, $100 plus like $75 for the plasmid. So $175 for 50 tests. And then plus the the probes from Thermo Fisher, which didn't work that great for me, or QuantaBio, they're not that expensive either. So I don't think it's cost. The qPCR machines are very expensive, like usually like $8,000. That would probably be the limiting factor. But if you can score one cheap off eBay, this one here was $850 and it came from a dairy. It's a fairly recent model year, but 2 of the wells don't work that great, so I don't use those. So I have 6 wells at work, but the open PCR machine, qPCR machine, was even cheaper. It was like $250. It was a bargain. So
in terms of reproducibility, what could be more or less like Yeah, what the technical things that you have encountered, or if there are any issues that if someone adventures to replicate it, what have you seen in during setting up with your experimental material?
Well, I mean, you have the negative controls, the positive controls, and you run them, and it's, you know, just look at the shape of the line that the qPCR gives you. I was an absolute beginner at qPCR. I didn't know how to do it at all. So I pretty much learned it from the CDC protocol, which is very detailed and has a lot of helpful hints in it. It goes on for like 40, 50 pages. But that was, you know, if you follow their own protocol, it's pretty good.
I remember during the beginning of the pandemic that there were like a flow of several types of protocols. I think one released by the Max Planck at the early times, the other one that became standardized by the CDC as well, and one by even the Institute Pasteur as well.
Yes.
Do you think it's any different? I mean, of what you have experienced so far. I mean, in order to think that perhaps people now are not so concerned about testing still, and maybe a little bit more on vaccination. But my question goes in terms that we're seeing waves, right, of COVID being increasing, almost like in different entering different seasons. So as the protocol has been depurated and anyone can access them, have you encountered or have the chance to compare them and found like what could be the advantages of using one part from one protocol or using one part from another protocol, that maybe you can make like your own Frankenstein protocol that could be a little bit more precise or anything? I don't know if you have explored that.
Well, there's because it's RNA you're dealing with, it's single-stranded, so you have to convert it into double-stranded. So you have a choice of using a one-step master mix or a two-step protocol. And of course, I would choose the one-step because it's simpler for me, and I would always take something simpler. So that's what I did. That's why I chose quantum bio. You know, the extraction step, you don't have to preserve it and play with it and extract it in a column or anything like that. You just take it raw and heat it before you start your PCR, and that denatures it enough that it's not contaminated and it works. And it's approved by the CDC for that, for resource poor areas. So I went with that. There are different tests. China has a test, Europe has a test, but you know, it's pretty much covers the same, you know, there's probably different primers. At first, the US primers did not work on one of the N proteins in the kit, and that kind of I screwed it up. And when I ordered my kit, I waited for a while because I don't want to take the kit out of hands of people that really needed it. But no one from our health department was ordering it, and they didn't have any capability. So I figured we might as well have to do it ourselves. But I think antibody testing is what we should be doing now, titer testing, to see who needs a booster or not, who has antibodies. That would be very productive, like the previous talk. More than, because people aren't testing and tracing now. They're just testing, but no one isolates anyone. They don't trace who their contacts were at all. And so they just go and infect other people. So the testing isn't as valuable as it would have been had people done the Chinese model, test and trace and isolate, but they don't do that here. No one's even wearing a mask now where I live. So it's kind of frustrating. So I think antibody testing is this, he who needs the booster the most.
I think Rachel was mentioning something really interesting when she was presenting Corona Detective. It was about the variant, right? That they expect that by redesigning the primers, they could achieve to have faster rate of polymerization and in consequence, a quicker response. During your experience about setting up in a different environment that is not the typical clinical environment, have you ever find out or have like questioned yourself if it is possible to also detect some of those variants in the population that maybe could be shifting from one place to another. I don't know if that's even possible at this moment.
Yeah, I think you could easily design your own primers and search for you. I mean, it's not, this qPCR machine is not limited. There's a whole bunch of other tests that the manufacturer sells for other different diseases. Like you push a button and it'll do it for you when they send you the kit. It's very simple to use, so it's not limited at all to this particular form of COVID. It can be used for any disease. You know, that's you can test that's genetically any bacteria, I guess, or virus. So it's very handy, but I think we're shifting away from the testing at this point because no one's isolating, unfortunately.
Okay, Xavier, I think, I don't know if we're already overlapping in time. If anyone else wants to add something to that or want to propose anything else regarding detection of COVID and using these tools, I believe this is a great moment to share also that type of information. I mean, it's amazing that you achieved to set up the whole thing, and maybe that could be replicated elsewhere. And this is a great experience that can be transferred to other labs or to other community labs that maybe are short in accessing testing. You know, like quickly giving context. I mean, here in Mexico, one of the most important limitations is actually accessing for COVID testing. It's still a big issue that we have one of the lowest rates for detection around the world. Even though that the city more or less have achieved to get like 45% of vaccination countrywide, the city maybe around 75, Mexico City particularly, or urban areas. In remote areas, I believe is when there is a big challenge of accessing to accurate testing. So yeah, I mean, it would be fantastic to be able to connect with those communities as well.
Yes, I mean, the high-tech solution, which is, you know, have the paper strip like the CRISPR Sherlock test for many different diseases. Again, that could be applied, and they could easily do that, but I don't know how they haven't, and they haven't scaled it enough yet. But any hospital or any university should have been testing with their qPCR machines for COVID in the beginning because no one else was. And I think the Broad Institute stepped up in Boston and did massive testing and converted their high-level research into testing, and a few others did as well, I think Ginkgo, but most universities did nothing, and they had the qPCR machine just sitting there doing nothing, which is inexcusable, really. I mean, they had the capability, and if you tried to buy a qPCR machine on the internet, it was super expensive because it was a huge demand. Even like a Biorad old model, it was like $5,000, and it was probably going to break right away anyway. Yeah, so yeah, I mean, there should have been more testing. It shouldn't have been left up to community labs or me or anyone else. It should have been easily available. It was unfortunate, and it's kind of like toilet paper. I mean, my God, how come we run out of toilet paper? We got to do this all by ourselves, really.
Right. What do you think about validation? Like, for example, when we start.
Real quick.
Oh,
yeah,
sorry.
there's a hand raised.
Oh, yeah.
Yes, but...
Oluwadamiola, do you have any questions? Go ahead.
Okay. Why do you think that there hasn't been and there's still no government buying and public buying of testing? Why the governments of the countries all over the world not have buying into testing and the public not also buying into testing?
I think some countries are. I mean, China certainly has. And yeah, but
I'm sorry about that. I mean the countries that have institutional democracy as their mantra. I'm not saying authoritarian countries.
Oh, okay.
No.
Well, Oluwadamiola, if you ask in the case, like for example, in Latin America, in particular in Mexico, I think is one of the most worrying feelings now that the system is not investing much on testing because it's affecting the public figures, and it has been that issue has been taken from several media. And unfortunately, the government that is running, at least in Mexico, where I had a chance to participate with many policymakers at the beginning of the pandemic, it was something that was not on the radar, not because they were not aware that was needed. I think it was not so good for their for the storytelling. You know, it was not something that they want to project over. And let's say that, well, Mexico still struggles with many economic issues, but in terms of healthcare spending, I think just a few years back, the infrastructure that we had before the arrival of this administration, I think it was strong enough to actually be prepared, or at least be in communication with other governments in the world that were experiencing similar bottlenecks. But from my personal experience, very, very personal experience, I can tell you that it was about the political narrative. It was totally in opposition to believe that when they just arrive in power, everything's going to be fixed up just immediately because people voted for them. And it's the most silly thing, I mean, honestly. And sadly, it was all the programs that were like doing this survey, this social survey actually, that for how this, because we have a lot of that in the rest of other remote areas in Mexico, like for example, Zika and the chikungunya virus and many other stuff. There was a program that was there as a surveying for continuously checking on the population that could be suffering that. And when this administration arrived, everything was shut down. I worked briefly in public healthcare administration, and it was terrible to see that very brilliant people participating on those programs, they were just fired at the beginning of the pandemic. So I believe this is in a very uncomfortable area between politics, more than actually more politics than the capabilities, the build capabilities that many countries have. I don't know if that answers your question, but I believe from the personal experience here in the Mexico healthcare policy makers interaction, this is what can I come to the table, and I hope it can improve a little bit more. But unfortunately, that was the main motive. It's because they were not looking good. Sad, I know.
Thank you.
Thank you, David. And I'd like to thank everybody for your presentations. But yes, also Daniel, thank you also for this panel. I'd like to thank everybody for their presentations, everyone's input. This has been an enjoyable this has been an enjoyable block, and I'd like to let everyone know at 7 o'clock, everyone should join the nucleus for a central talk. If anyone has any additional questions or any additional extensions of anything that's been discussed, please bring it to Slack. And I wish you all a fine rest of the evening and or day, depending on your time slot. Anyways, take care, everybody.